Hi, my name is Louis Bond. I'm the chairman of the RSVNet Foundation, and it's my pleasure on behalf of the RSVNet board to welcome you all here in this beautiful hotel here in Mumbai. Together with Leila, we will have a short introduction on the foundation and to welcome you all. Before we do that, I want to share with you, and this is a very special moment, I'll explain in a second, our vision and mission. It is our vision to support performance of high-quality research, to improve knowledge of RSV epidemiology, and to develop safe and effective therapeutic interventions, with the mission to connect global expertise and leadership in order to decrease the global burden of RSV infection, with the goals to advocate and create awareness of RSV and to combine knowledge and capacity required to enhance the development of novel RSV therapeutics. And for about 10 years, this has been our vision and mission statement. We never changed. This is what we are. This is what we do. And with the board, the entire board yesterday, we had a very lengthy discussion, and we felt it's time to change. Why? Because we have reached part of our vision. We are there. There are RSV vaccines. We have decided that it's time to change, that the development of vaccines is not enough. It's really about using them that needs to be incorporated. It's not about the development of vaccines, but using them. Not for children in some parts of the world, or pregnant women in some parts of the world, or older adults in some parts of the world, but all those who need it. So I want to make it very clear that RSVVW for the world is why we're here in India. So I want to shortly update you about the RSVNet activities, because we're very proud of that. We do science, we try to create awareness, and we perform educational activities. And very shortly, I will touch on them very briefly with one surprise. I hope you like that towards the end. So in science, we have received several large EU-funded grants, multi-million euro grants. Promise, rescue, clarity to decipher the burden of RSV, but also to develop new drugs for RSV, really translational research, IMI-funded projects. We have received private sponsor funding. We have 100 publications now, if you look it up at PubMed, more than 10 in Lancet with more than 2,000 citations. And we have a dashboard that you can actually look into at our website, and we think they are very useful. And you can see here, I'll use the other pointer. You can see here how well we're doing. So we try to create awareness with the voice of the patients. We have an RSV Awareness Week every year, with support of the Patient Advisory Board, and you can actually see using web-based search engine that we actually do make an impact. It is noticeable that we have an awareness activity, and this is our patient network from various countries. Several of the Patient Advisory Board people are actually here. If you want to talk to them, please approach them. They are the center of our work, and we're very proud that they're here, and we try to expand that to other parts of the world in the coming years. I'm pretty sure about that. So this is something that we really value. From the educational part, we have had a series of webinars. Probably all of you have at least seen one or two of them. They're well attended. The evaluation is superb. We have participants from all continents, and we feel that we get the best speakers. In general, we invite the authors of recent top publications to explain what they have been doing to facilitate, to help you understand the value of those publications. We're very proud to have done them. So this was the little surprise I was going to tell you about. We feel that with RSV vaccines reaching market, there is a lot of interest in being informed about RSV, so we have decided to develop a chatbot. So for all those of you who are under 40, I'm not going to ask whether you know what a chatbot is. For all of you who are above 50, I'm going to tell you that you should know what a chatbot is. And it's not ready yet, but I'm going to give you a trailer. This year, in 2024, we will have the first RSV chatbot for the general public, so everybody can be informed about RSV, and it will take a minute and a half to show you the next trailer. So this is a promise for you. This year we will make this happen. A lot of people, including some people here in the audience, are working very hard to make this a very high quality chatbot. And I'm going to ask you to contribute, each one of you. In the coming months, we need people who test the chatbot. So we are going to reach out to you, particularly people from different countries, different places in the world, and we're just going to ask you to invite you to use it before it's going to be online for the public, just to test it. Because testing a chatbot like this that uses, let's say, ChatGPT as a basis and expert knowledge to really have a good, solid scientific basis is what makes it better and better. So it's going to be a self-learning chatbot, and you'll hear more about that. So without further ado, I'm going to give the floor to Leila, who's going to do a special welcome here in Mumbai. Leila.
Thank you. So I said it yesterday, but today also, once again, welcome to Mumbai. And it's really nice and great to see so many new faces, but also so many faces that we already know from previous meetings. So we just wanted to start with, where is the slide? Oh, yes, thank you. We want to start with recognizing the fact that we know that for some of you the journey to Mumbai was not easy, and also the fact that there were some difficulties with some of the abstracts and the abstract notifications, and so we want to apologize for that. So on behalf of the team, it has been a very busy time and we have worked very hard, so just want to say sorry for that if you have experienced any inconvenience about that. And of course, the visa application wasn't easy. I know for some people it was very stressful time. So thank you very much for enduring with us. And it all worked out because you're here. So we have 400 participants here, and that's a really nice, let's say, numbers, but also really great that you all are here. We have people from 54 countries and from all the continents of the world. We were able to provide travel grants, 62 travel grants to researchers from low middle income countries and also young researchers. And it's really very fascinating. It's really nice to see all of you. A lot of you came yesterday to us and thanking us for being able to be here. But we want to thank you for being here and to share your knowledge with us. 191 posters, so there will be posters in the area across the hall, and it's open, so we have during the coffee breaks, we have time to go and see the posters. And as always, I've always said, RSVNet is a platform for all stakeholders involved in RSV research to come together. So here we have researchers, physicians, pharmaceutical companies, but also decision makers and like WHO, but also Gates Foundation, NIH, CDC, and PATH being all here. So acknowledgements and thanks to all the speakers and chairs of the conference. Very, very nice to have, I think, almost all of the speakers that we invited, they all said yes, and they are here. So thank you very much for your active participations in working with us on preparing the sessions, but also on sharing your work with us. As you can see on the top, you see Barney Graham. Unfortunately, he is not able to join us due to health issues, but he is passing his regards, and he's very sorry not to be able to be here. One more thing that I want to share with you is that we have, I think most of you who have been in RSVNet conferences, you are aware of the fact that we always have one inspirational speaker, and we want to inspire you with somebody who has achieved or who is doing something really inspiring. And this time we have selected Dr. Pawan Agrawal, and he's the CEO of the Mumbai Dabwala Association. And it's really, let me go to the next slide. It's really, I don't know, this is really something special, really special here in Mumbai, and he can tell the story, so I'm not going to do that. But they offer homemade, hygienic food at a reasonable cost, and they deliver it at the workstation, and also they pick it up. So there are 5,000 employees here in Mumbai, and if you have been able to go in the city, you see the traffic, right? And how busy it is, and also how big the city is. And it's really a big challenge. It may sound very simple, but it's not. And so he will tell you about, the journey of delivering of 200,000 boxes every day, and also getting them back. And they really work at a Six Sigma efficiency, and they have just one error in every 60 million deliveries. So that's really a well-oiled machine, and he will talk to you about it this evening during the reception. We will ask you to be here for let's say maximum of 30 minutes to hear his story. Of course, our sponsors, very, very big thanks to our sponsor. We would not be able to be here without the support of our sponsors. We would not be able to provide travel grants without the support of our sponsors. So big thanks, and I hope you keep supporting us in the coming years as well. So I already mentioned that Barney is not going to be here with us. We want to enjoy your time here at the conference and also enjoy your time in Mumbai. For some of you, you have already indicated to join us after the conference to go to do a tour in Mumbai and also a bit outside of the Mumbai. So looking forward to, to meet you there. In the meantime, I would say enjoy the conference, and I will give the floor now to our conference chairs. So we had 3, but we have now Heather and Joyce, who are here. Come on. Thank you.
But you've got to go. I think this one's. But it's got this one back. Okay. Technological challenges in India? No, maybe in South Africa. But good morning, everyone, friends, colleagues. It's really wonderful to be here. Wonderful to reconnect with this amazing community and also to meet new people who are also excited about the RSV world. What an incredible time to be coming together. As the reality of RSV being a preventive disease is here. We will hear a lot, and we heard yesterday some of the maternal vaccination or long-acting monoclonal data and overall results that are really making RSV a preventable disease and that are going to change the face of pediatric pneumonia. I don't have to remind this audience of the burden of pediatric pneumonia, 2 to 3 children dying every minute. And we know that RSV is a predominant pathogen. So this is an incredibly exciting time and really reflects the incredible work that has been done by people in this community and further over many years. I think, though, and Louis has stressed this over and over again, that one of the biggest challenges we're going to face is how do we get these interventions out to the children that need it, to those in low middle income countries, and how do we do this fast? We don't want to be waiting 10, 20, 30 years as we have for other vaccines. And I think the collective expertise and vision of a community like this will be critical to achieving that. It's been a great, great privilege to be involved in the organization and development of this meeting. The Scientific Advisory Committee, you can see, is really, it's humbling to be part of a group of people with the incredible expertise and the incredible contributions that they've made over so many years. And the program, I think you'll see, covers the latest developments in RSV research. There is a focus on low and middle income countries, India specifically, and I think the choice of this venue is very deliberate, and the ethical and equity issues, and a very diverse panel of speakers. Today, the sessions will focus on the global impact of RSV, on patient involvement and ethics, and on the young researchers' presentations. I'm going to hand over to Joyce to say the rest.
Yeah, thank you, Hita. Good morning, everyone. And for me, it's an honor to be here and to have participated in organizing this conference. And I would like to appreciate and thank RSVVW Foundation for asking me to assist in being a chair for this conference. So I'll move on to introduce the sessions for today. So the first session, that is of day 2, which is today, is on session 1, Global Impact of RSV. This will be chaired by Harish Nair and Vasha Potda. Session 2, that's patient involvement in and ethics. So this will be chaired by Louis and Taban. And then after that, we'll have our usual Young Researchers Tournament. So this will be chaired by Patrick Mnyoki and Mata Nunes. So for day 3, which is tomorrow, we'll have session 3, which is RSV clinical interventions and their global use. This will be chaired by Ruth Caron and Jacob John. Session 4 of day 3 will be implementation of RSV interventions. So the chairs will be me, and my co-chair will be Jessica Fleming from Perth. And session 5 will be RSV co-infection and long-term impact. This will be chaired by Hithaza and Renato Stein. So we'll have our day 4 sessions, which will be the last day for this conference. And session 5 or 6, that will be RSV vaccine immunology. So will be chaired by Tony and Ruth. Session 7, that will be lower middle income country health, economics and policy. This session will be chaired by Padmini and Neil. And session 8 will have global health challenges in RSV prevention, which will be chaired by Naveen. So at this moment, I would like you all to enjoy the sessions from today to the last day. And I would like to call upon Louis to introduce the keynote speaker. Thank you.
Wonderful. So I just want to point out, please switch on your cell phones during the next break. which will be lunch. So in the booklet it says we have a short, very short coffee break. We will leave that in order to have a nice flow. We started at 10 and we will have lunch after session 1. So it is my great privilege to introduce Ab Osterhaus. He has a very, very long CV. He has been the virologist at Erasmus Medical Center, Rotterdam, leading a big virology lab with more than 150 people. He established a company. He was the founding, or at least the chairman for many years from the SV sister organization of RSVVW. He's currently working in Hanover as a virologist, and as one of the RSVVW co-workers said this morning, it's obvious that you have a very long CV if you're a little bit older. And then I felt very uncomfortable because I don't have a CV like Ab Osterhaus. I'm also a little bit older. So I will not dwell on everything you have done because it would take an hour up, but you're going to tell us here about lessons learned from animal in infectious diseases. The floor is yours. I look forward to your talk.
For the nice introduction. I think I'm a little bit older than you, Luis, but you'll make it. So basically the title I was given, Lessons Learned from Animals in Infectious Diseases. And if I can get the first slide. Yeah. Lessons Learned from Animals in Infectious Diseases. Obviously, I'm only going to talk about viruses. And I'm barely going to talk about RSV because RSV is not a virus that recently spilled over. And I just wondered why I have been invited here, because this is all RSV people. And when I look through the posters and when I look through everything that's being presented here, it has very little to do with the things I'm interested in, although I've been doing quite a bit of work on RSV as well. And if you look here, then on the left-hand side, you see the human-animal interface. And through the ages, even if you go a million years back, we inherited quite a number of viruses from our ancestors that are being passed on, like herpes viruses, retroviruses, and what have you. But the horizontal transmission, and you see that here in all these different episodes, which I'm not going to mention, but there have always been opportunities for different viruses to cross the species barrier. And you see here, if you look at the names of these viruses, which will all be familiar to you, all these viruses, they come from the animal world, or they have representatives that are there. So without any further ado, when I entered the field, I was a veterinarian. I was in veterinary practice for some time and I got immensely bored. But what intrigued me was these enormous outbreaks of foot and mouth disease, avian influenza, and what have you. So basically, I entered in the 70s, and from the 80s onwards, you see all these different episodes here that happened over time. And many of these I've been involved in, and it started for me with HIV/AIDS, and all these different infections that you all recognize, they come from the animal world. So I think this is not going to stop. This is going to be there, and there's a lot of predisposing factors which actually allow these viruses, and new opportunities are being created all the time in our modern world, now that we are indeed in the Anthropocene, as we call it, with global warming, all these kind of things, which I'm not going to repeat, but basically that's what it is. And here you see also new bacterial diseases. You see, if you really want to do something on emerging diseases, you really have to go into virology, I think. So we can all agree on that. So basically the last one, and we start at the end, is Mpox, or monkeypox as we used to call it. And it was a disease that was locked up in Africa basically, and now in the last 2 years or so we have seen the spread of this particular virus, or one clade of the virus, basically worldwide. And we are quite relaxed about it. We don't speak of a pandemic at the moment. The reservoirs are rodents. My big worry is, because these viruses, they come back all the time, and my worry is that this virus now has spilled into local rodent population, so that we have a situation that we have had in Africa. So we're all quite relaxed about it, and we see cases and outbreaks every now and then, and I think it's a major thing for the future. And we already in 2006, we were worried about it. It was actually after the eradication of smallpox, we really said, hey, you have to be aware of all these pox viruses in the animal world. And definitely, if you look into history, you see all these things that have happened with different pox viruses. And basically, we did an animal experiment in monkeys, and you see here basically the recommendation of WHO is that if you have an outbreak, you have to vaccinate as fast as possible. And we actually showed that vaccination comes late, and there is limited protection of the current vaccines, as we know today, and antivirals do a better job, as we know. Well, then there is, of course, the list of WHO, and these are what they call the priority diseases, and we can all agree to that. And it's very difficult to predict what is going to be the next infection. And I think the most intriguing one for me is disease X. Yeah, if you would have told 15 years ago that there was going to be a coronavirus pandemic, nobody would have believed you. Yeah, so it is the lesson there is expect the unexpected. Yeah, it's definitely a lot going to come from the animal world, because all these things come from the animal world. I think we have to realize that we are just another animal species, and being exposed to all these different viruses. And of course we have better ways to make vaccines and the like, but we have to be better prepared. That's the message of today. It's interesting that even influenza is not on the list. Yeah, and I think it's one of the most likely ones to be a priority disease, because I can predict that we will have another influenza pandemic, and it may come soon. Well, let's go through the list. Well, basically we have Rift Valley fever. Yeah, and CEPI is now giving grants for big projects, and we are involved now in what is a phase 2 study here. We did a phase 1 study here on a new vaccine for Rift Valley fever, and that's a disease that comes from animals as well, and especially it's being transmitted by insects, but also during slaughtering practices and the like, you have outbreaks all the time. So we just heard about Uganda, there is an outbreak, in Senegal there is an outbreak at present. So these things are coming up all the time. So basically, and this is something that's going to interest most of you a little bit more than what I've been talking about so far. It's about paramyxoviruses, and now we call them also pneumoviruses. And in the past RSV, which you see here, RSV used to be a paramyxovirus, but because the genome organization is different, as you can see here, this is the group of pneumoviridae. So it's a special family now because of the different genome organization. And what I wanted to look into is this one here. We discovered this virus here, that was in 2001 in Rotterdam, in young children who actually developed the disease, and the physician said, the pediatrician said, oh, this is RSV. You virologists, you just don't know how to diagnose that. This is RSV. And as a matter of fact, it was not RSV. So we showed that this was a new virus. And if you look at the posters, I have seen quite a number of posters now where we have metapneumovirus as well. That's quite interesting, I think. And it is not a disease of children only. So it's quite overlapped. There's one poster I saw this morning which actually shows the differences in children between RSV and HMPV, and there's actually very little difference there. So quite interesting there. So the first thing we tend to do when we find a new virus, and that's one of our hobbies, to collect viruses. The first thing we tend to do is to prove the Koch's postulates, which we did here. So we infected monkeys and showed indeed that we could reproduce disease in monkeys with this newly discovered virus. So basically, if you look today, then as you would say for RSV, the risk groups, so those are the people with a malfunctioning immune system, but also if you look in older adults, and you can see it at some of the posters here as well, some of the presentation, it is in the same ballpark in older adults as RSV and influenza. So this is a virus that we did not know in 2000. We didn't know it. It was there for sure. But our diagnostic techniques have allowed us to identify the new one. And to come back to the subject of today, where does this virus come from? We did quite a bit of work on that, and on the sequences you see it has many different clades in the meantime here, and even a C here, and that overlaps with the avian metapneumovirus. So when we started to talk about metapneumovirus in humans, we initially thought this was a spillover from an animal reservoir, that the children in Rotterdam had been to the zoo or whatever. We did serology, and then over Holland, and then Europe, and worldwide, and then we saw that every child in the world over a year of age has had this infection at least one time. And you see here the RSVs here, and we have different RSVs, of course A and B, but we also have some of the animal RSVs. You see, if you look back, and we can do that now by following the phylogenetic tree, then you can see it was about 1800 that this virus spilled over from an avian reservoir into humans. So probably there was kind of a pandemic spread of the virus that we are quite familiar with today. So as I told you, one of my hobbies has always been virus discovery. Like collecting stamps. We collect viruses. It's quite an interesting hobby. And of course, I started mainly with animal viruses, but the red ones, those are human viruses. So even today, you will find new human viruses as well. And interestingly, so before 95, I was only doing animal work, and when I moved to Rotterdam, Erasmus Medical Center, so we started to look into humans as well. And even today, we identify new viruses. The little arrows here is another group of viruses. That's the morbilliviruses, and you're probably familiar with those. And you see it here. Again, you see animals. So we have identified morbilliviruses here in seals, initially an outbreak in the Netherlands, northwestern Europe, in Baikal. And so a lot of these infections which caused major outbreaks in animals. And then interestingly, this is not our work here, but then there were CDV, canine distemper virus outbreaks in monkeys, in macaques, in China. And that's interesting because there's a lot of cross-neutralization, cross-protection between the different morbilliviruses. And interestingly, as we are sitting here, we've all been vaccinated against measles, or we have had measles, the older generation, and so we would not be susceptible to CDV infection. But my prediction is, if we're really going to be successful in eradicating measles, which is high on the list, we're not very successful yet for several reasons. But if we are successful doing that, like in the pox viruses, we have to be careful because I can predict that there will be a spillover of an animal morbillivirus. And we did quite a bit of work on that. And it's interesting to realize that morbilliviruses have two receptors. They use two receptors. On the one hand, you have SLAM. That's the receptor that is on cells of the immune system, lymphocytes predominantly. And then you have the epithelial receptor. That's the Nectin-4. And interestingly, this is quite a ubiquitous one. And what we see is we have made in the lab virus of CDV and measles virus, which express EGFP. And so if you infect an animal, you can actually see the infected foci there. And what you see here, CDV and measles virus, they use the same SLAM. In monkeys this is. You see they use the same SLAM. But if you look at the tongue here, you see here you see the lymph nodes. They light up with CDV, but not the epithelial cells as you see with measles. So the idea is indeed, that probably humans are not susceptible to CDV yet, and we have predicted how many mutations it takes for the virus to eventually spill over, and we think that that's what happened in China in the monkeys. So that's an important thing. So whilst we were working on that, and it was Rory de Vries and Rick de Zwart in the lab who were working on that, we also could label the PBMCs, the cells of the monkeys there. And what we showed is when we know that in measles, canine distemper, that the animals get leukopenia. So the lymphocytes disappear, but not all of them. And it's mainly the memory cells that disappear. And I showed these data to Brian Grenfell at a meeting once, and he said, that's interesting. When we started to vaccinate against measles, and you see that here, you see then here, measles disappeared very rapidly in the UK, but also in the US. But interestingly, also the other childhood infections, they disappeared in principle. So they also went down. How can you explain that? We know that these viruses, measles is very immunosuppressive. And that ended in an enormous enormous number of papers actually that could explain at the end of the day how this immunosuppression due to measles takes place. And this is an experiment we did in the Netherlands. You can only do that in countries where they have a Bible Belt, so to say. So we took PBMCs from children before a measles outbreak. And we know that every 15 years, more or less, we get a measles outbreak. We got approval from our ethics committee. And as a matter of fact, we had all these PBMCs there from the children waiting for the next measles outbreak. And then the interesting thing is when we looked after measles infection in these children, what happened is the same as what happened in the monkeys. Yeah, so their memory cells disappeared. So they were naive again, and so susceptible to all these childhood infections again. So that led to quite a number of papers there, and I think it's quite interesting. Good. Well, now shifting gears, we have to talk about influenza here. Yeah, and it is actually already in 1997 that we, for the first time, identified an H5N1 highly pathogenic virus for chickens, in a child that had died in Hong Kong. We wanted to publish that in Nature. Yeah, and as a matter of fact, with the comments that we got back from the reviewers was, you don't know your textbooks, These viruses do not go from birds to humans. And this kid had been to a marketplace and where there were outbreaks of this particular infection. And it took until 18 people became infected, 6 of whom had died, that we got the paper published eventually. And then, interestingly, we looked into it, but this virus did not spread from human to human. So that was comforting for the time being. But if you look over time, this was 97, and then we have seen repeatedly outbreaks of the H5N1 highly pathogenic avian influenza virus infection. And you see here, case fatality rates are quite high. Yeah, and this virus, this does not transmit from human to human yet. Yeah, and we were quite interesting, why not? By the way, this morning, again, I saw in Cambodia, so we see people dying from these infections, although it's sporadic. Although, in the meantime, so what we have seen for the H5N1 infection here, that it also affects other species from the beginning. And this was in cats, we saw it, and we speculated if it is in cats and other mammals, it could adapt to mammals and then eventually spill over to humans and cause a pandemic. The interesting thing is, we predicted more or less that the next pandemic was going to be H5N1. Whatever you predict in flu is not going to happen. It became an H1N1 from Mexico, yeah? So the swine flu virus. Well, anyway, we were wrong there, and Ron Fouché in the lab did quite some interesting experiments here, where we took, just to show what this virus would need to become transmissible from man to man, from human to human. And we did that in a ferret model, and as a matter of fact, we took a molecular clone of the virus here, and this one was not transmissible. You see it here on the right-hand side. This virus was not transmissible from ferret to ferret. We have a special system with a cage where we blow air over it, and there's a barrier here, and so the virus can only transmit from left to right, through the air. And as a matter of fact, we passaged the virus 10 times over the upper respiratory tract of ferrets. Still, it did not transmit. Quite interesting. Then we helped the virus a little bit by applying a couple of mutations there. And this virus did not transmit either. These are mutations that do occur in seasonal influenza viruses. And then we did the same trick again, and surprise, surprise, we had the virus here that indeed transmitted from ferret to ferret. And the ferret is quite comparable to humans in terms of its receptor distribution. So we said basically we have made a pandemic virus. Of course, that created some discussions whether you should have done it or not. Well, that's a completely different discussion. We had all the permits we needed, and we did it anyway. But and as a matter of fact, it boiled down to not more than 5 mutations in the virus that were needed to create a virus that would be transmissible. Well, if we go on, and interestingly, the work we are doing in seals, and I'm a little bit hooked to seals, it's quite an interesting species. We saw an outbreak of another avian influenza virus, this H10N7, and it went here from Denmark, so along the coast, spilled over to the UK, and it killed about 20% of the seals. Initially, it was stuttering, And gradually the virus adapted and it spread like wildfire, and then it died out. But we had the different viruses. We had swabs from the animals at the different moments, and we looked here, and it was Ron Fouché in the lab again who was doing that. We actually this was an experiment of nature. A virus that was barely transmissible adapted really in the seals over time to become very transmissible. So I think these things are happening in nature as well, and we were quite interesting. And now we have a pandemic ongoing, or it's not a pandemic, it's a panzootic, we call it. Yeah, it is actually an infection in birds. Yeah, H5N1, still H5N1, it's not exactly the same virus. A lot of birds are dying. Yeah, so terns, you name it, all kinds of birds. And it started here in Europe. Yeah, and the virus, it comes from Asia, here in Europe, and usually it was only in the winter time, but now we have it all year long. And the interesting thing is it spilled over also into the Americas now, and you see it here all the way. So this is, and it's now in Africa, it's all over the place. Interestingly, it's not yet in Australia. So we cannot, it's not a real pandemic yet. But even more worrying at the moment is the facts, and if you look here at the right-hand side, these are all mammals, terrestrial mammals, aquatic mammals that are being infected with this particular virus. And we also see occasionally outbreaks, meaning there is probable spread from animal to animal. These animals are being infected from birds initially, and either by eating the birds or eating their excrements. And the question is, of course, can this cause a pandemic? I'm not going to predict that H5N1 is going to be the next pandemic, because I may be wrong again. But basically, it causes enormous problems in the poultry industry, but also there's massive die-offs at the moment of sea lions, for instance, in South America, in Chile. So it's basically, we see it now in all these different terrestrial species, but also in the aquatic species. We see this happening all the time now. And we did quite a bit of work on it recently. And well, having worked in this field for a long time now, all the circles is viruses that we have identified over time. So you see in these aquatic mammals, we quite often see these outbreaks. This is interesting. Influenza B virus. You probably have learned that influenza B virus is a human-only virus. And we saw here, this is probably a spillover from humans to seals. So this happens, but all these other infections, and so you see two infections with an H5N8, it's another subtype, yeah, and also with an H5N1 in some seals from Germany, but also some Dutch and German seals. And the interesting thing is these seals do not get a pneumonia. They die before they get the pneumonia from an encephalitis. And you wonder, how could that be? And we think, yeah, and we have some evidence for that they actually swallow the material, yeah, either they eat the chicks or they get the poo in their gut, and then the virus goes through the nervus vagus into the brain directly, and they die from a massive encephalitis before they can actually develop a pneumonia. So we see this happening all the time, and interestingly, the highlighted viruses are the seal viruses, but all the other viruses are bird viruses. You can see these seals are in the middle, the seal virus are in the middle of all these bird viruses. So what we see is, what we do, we sequence these viruses in the brain, yeah, and what we see is there's a number of mutations, as you've seen before, that are suspect of the virus to become transmissible. And now, for instance, we already see that one of the mutations here, yeah, we find in the brain of the seal. And we just wonder what's happening with all those seals in Chile, in South America, the sea lions and the like. Okay, well, shifting gears again, but still staying with influenza. During the pandemic, the swine flu pandemic, we looked in Erasmus Medical Center in the hospital, and we looked at immunocompromised individuals here. And this is the highlighted areas here. This is therapy with oseltamivir. And what you see, the mauve viruses here, they have developed the escape mutation here, H275Y, and this makes them less susceptible or unsusceptible to oseltamivir. And you see that happens after some time. And so we thought it might not be a bad idea to develop a model, an animal model. So what can we learn from animals? And as a matter of fact, we developed a model in ferrets, and you see that here. These are normal competent ferrets. When you infect them with this virus in the nose, and you see excretion of the virus here, and all these animals survive. when we immunocompromise the ferrets, and this is according to a regimen that is similar to what we use in renal transplants, and those were the main patients you saw on the previous slide, then you see they all die. Yeah? You see they all die. But then if we then treat them with oseltamivir, interestingly, half of them survive, half of them die, but you see they all develop the same mutation within 2 weeks. Yeah, so this is a nice model that we're using also for testing other antiviral strategies. And interestingly, we have now also used this, and for this audience might be interesting, also for RSV. We use the same model here, where we use immunocompromised ferrets, yeah, and we infect them with RSV, and that works quite well. But we have some alternative systems there as well. So we use organoids, and we use Ali cultures as well, and PCLS. So we are doing a lot of this animal work. And as a matter of fact, we learn a lot from those model systems. Well, in the meantime, Martin Ludlow in the lab, he has made a series of contemporary molecular clones of RSV-A and RSV-B, and you see them here, and they have these marker proteins in, and you see that here this is for A and this is for B. And we've used these viruses now, and they light up. You know, if you use a UV lamp, you can see actually, as I've shown for the measles experiments, for the morbillivirus experiments, you can see indeed where the virus is. And we use this virus. We have these viruses now, these molecular clones, and we can just put the mutations, the resistance mutations and this is for palivizumab, for instance, we put them in, and then you see that the inhibition of the virus, and you see here, they break through, whereas in the case of the original viruses, it's not so. So this is a system where we can quite rapidly manipulate and look for resistance there. We've done these experiments in the meantime also in animals now, and it works perfectly. You know? So this is quite an interesting system to have. Well, shifting gears again, now going to close off, going to the coronaviruses. So when we had the SARS coronavirus outbreak, it was interesting to note that we joined forces with a number of groups. And as a matter of fact, what we have here four publications that appeared more or less simultaneously. And this was the working group. We all worked together. All the authors are on all the papers here. And we actually showed that this was caused by a coronavirus. And basically, initially, when we identified the viruses that were in the patients with SARS, there was a coronavirus and there was the human metapneumovirus. And all these people on board, you know, they were medical people, and they said, well, it cannot be the coronavirus because these are wimpy viruses, and they did not realize that these viruses in the animal world cause massive disasters, like in cattle, in birds as well. So, as a matter of fact, then in the monkey experiment, we proved the Koch's postulates there and showed it was the coronavirus and not the metapneumovirus that was at the core. So we used that system, yeah, also to test, in this case, different medication. You see an example of interferon using pegylated interferon alpha here that worked pretty well in monkeys to protect them. And at the end of the SARS-CoV-1 outbreak, so basically this was used in Canada, for instance, but it wasn't necessary anymore because the non-pharmaceutical intervention was sufficient to stop it. Well, we developed different other model systems there. I'm not going into details there, which we could use is cats, ferrets, and we know in the meantime now the virus came from bats, you know, and then spilled over to civet cats to get into humans. 10 years later, after SARS-CoV-1, we had the MERS outbreak. Middle East Respiratory Syndrome coronavirus was there. We discovered that particular virus in the lab because we got samples from Saudi Arabia. And we have Dr. Zaki here. He is a medical microbiologist, and he noted one patient, this is the index patient, yeah, an elderly man who had died, and he contacted us, sent samples. He said, it must be one of those paramyxoviruses or whatever you're working on. See if you can find something new there. So we found, indeed, yet another coronavirus. It used a different receptor, yeah, yet another coronavirus. And as a matter of fact, the people in Saudi Arabia were not too fond of that it was actually found there. And they were in kind of denial. But we saw the virus simmering here. We have outbreaks here, an outbreak in Korea as well. and basically this was a virus that was quite important as well. So we were very quick on the ball there. We showed by looking at antibody profiles that the dromedary camel was the culprit. It actually spilled the virus to humans from the bat reservoir. We basically, in a farm, we could show that the virus that was in 2 people who came down with the disease actually was identical to the virus that was in the camels there in the upper respiratory tract. And Stalin Rai in the lab, he is now back in India. Yeah, so he actually he identified CD26 as the receptor of the virus, which is quite important. It's interesting to realize we could do this in a couple of weeks. Yeah? In 2-3 weeks, we had the whole thing fixed. And if you remember, when we were working in the old days on HIV, it took 2 years before the virus was identified, and another 2 years before we had the receptor. This was 3 weeks altogether. For SARS-CoV-2, yeah, it took a couple of days. Yeah? And also the receptor, well, of course they had an advantage there. So we, knowing the receptor was DPP4, CD26, and you see making a phylogenetic tree of the interacting part of the receptor, you can also predict more or less which animals are going to be susceptible. And we could already see that, for instance, the ferret that we normally use would not work. Yeah, and so we did, and the monkey would work, of course you see that here. And as a matter of fact, we did a couple of experiments. We produced some vaccines, and we had a vaccine based on the MVA. So it's a vectored vaccine, similar to what has been used for SARS-CoV-2. We did an experiment in the camels, and I can tell you an experiment in camels under BSL-4 conditions is not simple. Yeah? So these animals, they bite, they kick, they do whatever God has forbidden. And you start with small animals, but they get very big. So it's not like a BSL-4 experiment in mice, I can assure you. So we did that in Barcelona, and we were quite successful, the vaccine. What I can tell you, this vaccine, as a matter of fact, CEPI funded now, has been through the phase 1, is now going into phase 2 as well. Similar to what we have shown for the Rift vaccine. Well, you see here the different origins of the different viruses we know in humans. When I started when I came into the corona field, and by the way, I did my PhD on the coronavirus of the cat, which is also causing a deadly disease, then there were only 2 coronaviruses in humans. Yeah, and it's the 229E and the OC43. Now we have quite a few more, and there's an enormous discussion, of course, where this SARS-CoV-2 came from. And I'm not going to speculate on that any further. We know originally it came from bats, but how it got from bats to humans, we don't know. But again, it's coming from the animal world. You know? So I think that's important. All these new things, they come from the animal world. So basically, again, when we say, with all the efforts we did to develop vaccines, antiviral strategies, etc., we used animal models. And I'm not going into too much detail, but so you see, well, what we did for SARS-CoV-1 was repeated here by us and others for SARS-CoV-2. And you see here we go from mouse to hamster. Well, these are transgenic mice with the ACE2 transgenic, to ferrets, to monkeys here. And a lot of experiments were being done. So we have set up the models. And one of the things we did, whilst we were seeing all these new variants of concern, we tested them in hamsters as well. And you see here replication of the virus in the lungs, in the upper respiratory tract, weight loss. And we could actually follow what was happening in humans was actually very similar to what we saw happening in hamsters. And they provided good models there for testing intervention strategies. So just an example here. The problem with the monoclonal antibodies that we developed, we were very, very fast. Yeah, we made them with the group in Utrecht and Rotterdam together and tested them in hamsters. And by the time we wanted to start clinical trials, we had another variant again. So that happened all the time. But still, we learned a lot. And you see here one antibody here. Yeah, it's against the Omicron variant. And you see here the isotype control. This actually palivizumab we used here. And then in the lungs of the hamster, you see there is complete protection there with this antibody, like with that's an antibody that we produce. But we have a whole series of them. But these hamster models were quite instrumental to see all this. Well, now we shifted gears as well, and it was actually interesting in Louise's introduction that what we need to do for the future is also to see, well, for instance, when we talk about the monoclonal antibodies that we want to make, that we can produce them locally at a lower price. So we are looking into different alternatives. So I was on the board of Protein Sciences to actually use insect cells instead of mammalian cells. So that vaccine is on the market for flu now. But now we are using a fungal system as well. And you see that here. And here we have compared one of those, it's actually the same antibody here, produced in the fungal system and produced in the mammalian system. We've compared, and it gives complete protection in hamsters, and we just performed a monkey experiment as well. So we could show that we can produce these antibodies indeed in the fungal system at a very low price. Cost of goods is very low, and we still have to do all the mathematics there. But this is just to show that there are alternatives there. Similarly here, again, in the same system, we produced a candidate vaccine on the basis of the RBD, and we showed in hamsters, you see here the antibody production here in vivo, and we also showed protection there. So basically, we're very much interested also into in aging, and this is some studies that we did with Guus Rimmelswaan in the lab, where we looked at senescence markers here in humans, and then looked at the correlation with T-cell responses, antibody response, and we have done similar thing in old macaques, and then for SARS-CoV-1, and we are doing that also for SARS-CoV-2. So we learn a lot from the animal models, I should say. And just to finish, Mr. Chairman, just to finish, it's interesting, of course we were quite interested in you've seen all these spillovers to humans, and also, so all these viruses that come from the animal world, but there's also, in the other way around, at the very beginning, we had indications in the first wave of SARS-CoV-1, SARS-CoV-2, that we had dogs and cats infected. So we did an epidemiological study over Europe, and what you see here is that even after the first wave, about 4% of the cats in Europe were seropositive for this virus, and before they were all seronegative. So that was a spillover to cats. And obviously there is little risk there because the owner was already infected. This is from owner to cat. But what is even more disturbing, I would say, if you look at the number of animal species here, and you see this is a year ago, but 15 mammalian species, in 50 mammalian species, the virus is present, and even in some of them it's circulating. So we may expect that this virus is out there. I showed you that for pox virus, I showed you that for the morbilliviruses. So my message here is indeed that these viruses might come back. And with all these predisposing factors that we have today, I'm afraid that we are going to face many more of these outbreaks in the near future. Well, my conclusions, Mr. Chairman, the lessons we have learned. So I think all major human viral diseases continuously come from the animal world, either from terrestrial, either from wildlife or from domestic animals. And wildlife, then I've shown you terrestrial animals, aerial animals, flying birds, transmitting it to chickens. So going then to domestic animals. And the other thing to realize is that animal models have been shown to be crucial for the testing of our new intervention strategies, obviously. Although, and I didn't spend much attention there, there's an enormous push at the moment, of course, to go for in vitro system, alternative systems, which cannot replace this altogether. But it's important to spend a lot of time on that. So basically, this is to thank our group. And you see, we have in the meantime, I didn't realize that we have already 4 people from India working in the lab. So I'm not sure if this number is going to increase in the future, but it's quite a mixed group. And you see here, these are two students from Stalin Rai, who you've seen before, and here Guus Rimmelswaan, Martin Ludloff, whom I mentioned, and we get funding from a lot of different groups. Thank you very much.
Of interest is respiratory viral epidemiology. I've done some work around RSV as well, and I think, we've heard all of yesterday the importance of RSV, particularly in young babies as well as in older adults. But there were some pediatricians from India I was interacting with, and they said, we see a lot of RSV, but we don't see a lot of deaths from RSV. So and you've said that there is a lot of deaths. What is it all about? And I was wondering where that comes from. So we all know that pneumonia is one of the leading causes of child mortality. Globally, about 700,000 deaths annually from pneumonia in under 5 children. And 1 in 4 of those pneumonia deaths occur in India. So this is one of those countries where there's a high mortality burden from pneumonia. Then what is the role of RSV in child pneumonia worldwide? We know that about 1 in 3 of the pneumonia cases worldwide is attributable to RSV, and this has been shown in several studies now. And as far as deaths are concerned, about every 2 minutes, 5 deaths occur from RSV in under 5 children. And if you were to look at all-cause mortality in under 5 children, 1 in 50 deaths in under 5 children worldwide is attributable to RSV. And in under 6-month-old children, the burden where it's most severe, 1 in 28 deaths from any cause is attributable to RSV. So it's one of the leading causes of child mortality outside pneumonia and outside malaria. The other important thing to bear in mind is a lot of us are clinicians, we're seeing deaths occurring in hospitals, but do bear in mind that in pneumonia, the majority of the deaths, even now, in low and middle income countries, including in India, they occur outside the hospital. They are occurring at home. And for every 1 child who dies from pneumonia in hospital, there are about 3 children who are dying at home. And similarly for RSV, for every 1 child dying from RSV in hospital, 3 children are dying at home. So it's really an important cause of child mortality, and through the sessions today, we would be highlighting this importance of RSV, particularly in terms of impact in low and middle income countries. I now hand over to Varsha to introduce herself and introduce the first speaker.
Good morning all. I'm Varsha Poddar. I'm a scientist at National Institute of Virology, and I'm heading National Influenza Center of India. My interest is on influenza and other respiratory viruses. Mainly we work on molecular epidemiology of respiratory viruses. Currently working on evidence-based research activities for influenza and respiratory viruses, which includes RSV. For improved public health actions, basically, I'm a part of WHO RSV pilot study as well as genotype group. And mainly we are actually ICMR NIV Pune is a apex laboratory for ICMR, and coordinating this nationwide influenza and RSV surveillance, which will, I hope that in future we will able to generate more RSV related data, and we will come up with the future action plan for that. Thank you. Now I will introduce the first speaker, who is Dr. Ashish Sathav, who is the director of Mahan Trust, Dharni, Amravati area. Dr. Ashish is basically worked in a tribal area of Meghat since last 25 years, and he has treated more than 1 lakh patients, basically saved more than 5,000 lives, and we have done together a study for the RSV disease burden with Gates Foundation funding, and we have published 2 major kind of findings for the community-based as well as the hospital-based severity for RSV disease. With that, Dr. Ashish has received many more prestigious awards. Total 74 awards he has received for his work. and he has contributed in government policy change and that was benefited to more than 10 lakh poor tribal peoples. So with this introduction, I hand over to Dr. Ashish for his talk.
Thanks, Varsha and Harish. My topic is RSV burden in India. Mahan Trust has received grants from Bill and Melinda Gates Foundation via University of Colorado, Denver for this study in Meghat. This was our area, Meghat, situated in this forest of Maharashtra, northeast part of Maharashtra, 800 kilometers from Mumbai. This is our team, Dr. Eric Simois from USA, Dr. Nitin Vairakkar, Dr. Dani, Dr. Varsha Poddar, Mandip Chadda and others from Mahan Trust. Mahan Trust is providing medical services to tribal area of Meghat since last 25 years. The various programs are home-based child care program and community-based management of severe malnutrition, mortality control program for economically productive age group, well-equipped multi-specialty hospital, then Umang de-addiction program, sustainable nutrition programs, blindness control program, RSV study, minimal invasive tissue sampling study, counselor program for strengthening of government hospitals, then TB and pneumonia studies, policy change through advocacy, research and PIL. It has benefited around 1 million people across the state of Maharashtra. As speakers have told, RSV has high mortality and morbidity across the globe. It is one of the leading cause of mortality and morbidity in under 2 children as well as in under 5 children. In India, RSV is the leading cause of respiratory tract infection, especially lower respiratory tract infection. It contributes to around 25% of all viral pathogens, respiratory pathogens, while in the globe it is the second leading cause after rhinovirus. When you compare the incidence of RSV in different parts of India, in central India, in the community, RSV contributes to around 13.59% of total lower respiratory tract infections, while in North India it is around 6.86%. When we compare the contribution of RSV in LRT and India, it's around 10.94% in the age group of 0 to 23 months. When we compare the hospital-based study, the prevalence of RSV in the lower respiratory infection in East India is around 10.3% in the age group of 0 to 23 months, while it is 2.4% in the age group of 2 to 5 years. In West India, the prevalence of RSV in hospitals is around 22.64% in the age group of 0 to 23 months. In Central India, the prevalence is around 23.38% in the age group of 0 to 23 months. In North India, the prevalence of RSV is around 15.25% of all lower respiratory infections. When we combine, while in South India, it is around 28.5%. When we combine all the studies, hospital-based studies in India, RSV contributes to around 16.85% of all lower respiratory infections in the age group of 0 to 23 months. The prevalence, when you compare the prevalence of RSV-A versus RSV-B, 5 studies has shown that RSV-A is more prevalent than RSV-B, while 2 studies has shown that B is more prevalent than A, and 2 studies has shown that both are in equal proportion. This RSV has very classical seasonal trend. It is more commonly seen during rainy season when the precipitation is high from June to September in India, and some part, the second peak is winter, late winter, that is December and January. In 2016, the RSV-A was more prevalent, while in 2017 it was replaced by RSV-B, which continued in 2018. When we compared this data with our study in Central India, we found that RSV has biennial variation. It was more common during 2016, 2018 and 2020, while in 2017 and 19, the RSV was replaced by HMPV. It was more common during again September, which was rainy season with very high precipitation, and more common again in December and January when the temperature was low, which was winter in our area. Before COVID, RSV-B was more prevalent. It contributed to around 9.15% of the total lower respiratory infections, especially severe pneumonia cases in the children in the age group of 0 to 23 months, which reduced to nearly 0 after COVID, while RSV-A it contributed to around 3.05% of total lower respiratory infections before COVID, which increased to around 42% after COVID. Similar situation was for deaths. RSVB contributed to 3.4% of total lower respiratory infection deaths in Meghalaya before COVID, while RSVA contributed to 2.31%, which increased significantly to 10% of total pneumonia deaths in under 2 children. The various diagnostic methods used in various studies of India, it was mainly real-time PCR provided by National Institute of Virology, Dr. Varsha Poddar and team. The sensitivity and specificity was from 98.8% to 100%, while other studies mainly use multiplex PCR, real-time PCR with sensitivity and specificity in between 90% to 100%, while some few studies use viral cultures also. When we analyze the data of all severe respiratory tract infections, especially pneumonias, the mortality rate per 1,000 child year was around 30.1 in the age group of 0 to 5 months, which reduced to 2.5 in the age group of 6 to 11 months, and again to 0.9 in the age group of 12 to 24 months, with community to hospital ratio was around 2 in the age group of 0 to 5 months and 2.1 in the age group of 6 to 11 months. RSV severe LRTI mortality rates per 1,000 child years in India was around 3.3 in the age group of 0 to 5 months, which reduced to 2.1 in the age group of 6 to 11 months and 0.7 in the age group of 12 to 24 months. Here again, there was more community mortality as compared to hospital, with community hospital ratio of 1.4 in the age group of 0 to 5 months, which increased to around 4.3 in the age group of 6 to 11 months. So there are definitely more community deaths as compared to hospital. When we analyze it further by age, we found that in the age group of 28 days to 90 days, the rate of RSV positive severe LRI was maximum. It was around 54.9 per 1,000 child years, which contributed to around 14.5% of all LRI in our area. In the age group of 91 to 180 days, it was around 46.9 per 1,000 child years, which contributed to 13.7% of all pneumonia cases, while in the age group of 183 to 365 days, the rate was 43.5 per 1,000 child years, which contributed to 15.2% of the total pneumonia cases. So we collected nasopharyngeal swab from 3,494, which was tested. Around 13.9% were tested positive for RSV in NIV. When we compared the mortality rate of lower respiratory infection and RSV, we found that the maximum mortality due to RSV was seen in the age group of 7 to 28 days. It was around 2 RSV per 1,000 child years, while it reduced slowly to around 0.4 in the age group of 28 to 90 days and 0.2 in the age group of 91 to 182 days. And overall, under 2 children, the RSV mortality rate was around 1.1 per 1,000 child years. There was very classical seasonal variations for other lower respiratory infections in our area, especially severe LRTI. It was more common during rainy season in the month of September when the precipitation was very high. And again, the second peak was in the end of December and first week of January when the temperature was low. When we compared this severe RSV LRTI rates in community in terms versus preterm children, we found that during the first year of life, the preterm babies has much more incidence of this severe RSV LRTI as compared to term infant overall in both hospital and community, as well as much more in the community as compared to hospital. And the rate was highest in the third quarter of first year of life. Conversely, it was almost the lowest in the first 3 months of life. You can see here the rates of preterm and full-term severe and very severe LRTI with RSV infection was much more in community as compared to hospitals. When we calculated the incidence rate ratio of full-term and preterm RSV LRTI in India, we found that during the first year of life, the preterm babies has much more incidence IR than the term infants in overall and in the community, especially in the community. It was maximum in the preterm babies. The incidence was around 26.3 in the age group of 0 to 180 days with IRR of around 1.66 as compared to full-term children. And during the RSV season, the incidence increased to 45.9 in the age group of 0 to 180 days and around 52 in the age group of 0 to 270 days. And the incidence rate ratio as compared to hospital was around 1.77, which was statistically significant. When we analyze further, we found that all of the RSV cases in the preterm group, they presented with very severe lower respiratory tract infection as compared to only 52% of the full-term infants. While for RSV B type, the proportion of very severe RSV associated LRTI was 44.8% in full-term cases and 52.8% in preterm cases. When we compared the monoinfection versus co-infection, we have found that the lower respiratory tract infections has much more RSV and much more co-infections with other viruses as compared to upper respiratory tract infections, and the p-value ranged from 0.016 to around 0.001. When we analyze the nutritional Z-score of severe RSV LRT cases versus control non-RSV lower respiratory tract infection, we found that the proportion of infants and young children with severe acute malnutrition or severe underweight increases at the time of RSV infection. This increase in children with wasting and underweight declines post-infection but remains at higher level as compared to the non-RSV lower respiratory tract infections in the control population. There was risk factors for RSV in central India through our case control study. The male, they are at more risk with odds ratio of 1.3 with p-value of 0.017. Mothers who were not employed, they were at higher risk with odds ratio of 1.3 and p-value of 0.021. The families who use kerosene, their children were at high risk with odds ratio of 5.8 and p-value 0.08. While those who used water from common source, community tap, they are at more risk with odds ratio of 1.3 and p-value of 0.022. And families, poor families from rural areas, they were more at risk with p-value of 0.004 as compared to wealthier people from cities. Other risk factors in the rest of India are family history of atopy or recurrent wheezing, lack of breastfeeding, smoking in the household, longer stay in hospital, viral load, escape from neutralization, low antibody titer, genetic polymorphism, low socioeconomic conditions, low parental education, exposure to tobacco or industrial smoke, male preponderance, and malnutrition and small for gestational age. We should support vaccine development and public health measures to minimize severe respiratory illnesses, alleviate healthcare strain, mitigate economic cost, prevent secondary infections, and safeguard the vulnerable poor population. To conclude, RSV has substantial burden on public health affecting vulnerable population. The burden of hospitalization during peak season is significant. The cost of medical care and lost productivity highlights the urgency of preventive measures. Collaborative efforts are needed in research, vaccination, and public health interventions, which are very crucial. RSV has highest prevalence, 26% of total viruses as compared to other respiratory viruses. South India has maximum prevalence as compared to East India, and it was more common during rainy season and winter, and the proportion of all very severe lower respiratory infections and severe pneumonia is higher in the preterm group with odds ratio of 1.66 with p-value less than 0.001. Mahan Trust is planning to conduct clinical trials or effectiveness studies for RSV products, to conduct trials on RSV monoclonal antibody or RSV vaccines for pregnant women in Meerut in future. I am very much thankful to all of you for your kind attention, and to the organizers and the chairperson and co-chairperson Harish and Varsha for their support. Thank you.
Harish. Oh, good, they're up. So I'm not going to focus so much on the burden of RSV, but I'm going to focus more on how we've gotten the data we've got at this point and what we should think about as we go forward, because now we're in a new era. We have a vaccine or several vaccines that work. And the question is, how are we going to employ those, and how are we going to study their impact over time? And I think this is important because it's not going to be as simple as one of the slides that was just recently shown, where it was almost magical. You give the drug and the disease goes away to a very large and obvious extent. And it's going to be much more difficult in the older population to get that kind of data. And these are my disclosures. So I'm going to sort of go through where did we start, where are we now, and what do we really still need to know that'll help us. So in the 1980s, this is sort of where we were if we were talking about looking at RSV in older people. It was sort of a pipe dream, a mirage. Maybe it really didn't even exist. There were very few papers on the topic, and almost nobody investigating this other than a few people who were interested in nursing home outbreaks of RSV that were seen as sort of a curiosity. But over the years, the number of publications started to pile up, usually in small journals originally, and then larger and more impressive journals as time went on. And you can see on the little graph on the bottom, the number of publications if you were to search for RSV in adult populations. And you can see the number has increased quite dramatically over the last 10 years. So in terms of analyzing some of the data that has been generated to date, and then think about it as we move forward in terms of the epidemiologic data, the impact data of RSV that can be generated, I want to think about a couple of types of studies that are done and then some of the factors that you want to consider when you're analyzing the previous data and when you're thinking of planning studies in the future. So I'll start with prospective cohorts, especially outpatient cohorts, and this is one of the areas where I think we have the least amount of literature. But it is the best way to do the studies. Unfortunately, it's expensive, time-consuming, and takes a long time to get the data because you're going forward prospectively. But it allows you the evaluation and presentation of medical care. And you can do this either with a cohort study or you can do this by camping out at sources of medical care, such as offices, emergency room, and hospitals. And although it's not fully prospective or in terms of a cohort study, it does allow you to really describe a lot of the clinical features, and then in the long term, if you follow them long enough, impact data. As far as retrospective studies, they have been very useful, and they can take standard of care testing that's done, which is going to be very variable from place to place. Database diagnoses suffer from standardization of data input, who's tested, which syndromes and symptoms are included in these analyses, and so you need to think about those things. And then other important considerations, as I mentioned here, the case definition that you use, diagnostics, and we will again focus on that a little bit here as I go forward, because I think this is important and something that could be quite useful in getting more data. And then also thinking about the timing of evaluations, that is it year-round, is it seasonal? This has become a bit of an issue during the COVID pandemic, obviously, and it does appear like this may not be a persistent problem. So just looking at some old data and some new data, using very standard technology such as PCR and serology. And these are two studies, and I think we showed these yesterday, or three studies actually, two in the chart. One is a 4-season study from Rochester, which found about a 5.5% incidence, or 5.5 number of cases per 100 individuals followed in a cohort study, and this is in people over age 60. And then a 2-season study done in 3 countries in Europe, very similar methodology 20 years later, coming up with really very similar numbers. And I might point out it used PCR and serology, and that serology increased the yield by about 30 to 40% in both of these studies, and that about 10% of these would be classified as asymptomatic or essentially asymptomatic enough that the subjects did not report an illness, but they were seroconverted during the season. And then a more recent study that was a 1-year study from the Mayo Clinic, using just PCR, finding a rate of about 2.5%. So that gives us using these techniques, a pretty good incidence in 2 or in 3 studies that are cohort studies, time-consuming, and require a lot of energy to do. So a conservative estimate might be anywhere from 3 to 4% per year. The prospective studies where you look at incidence of medically attended disease at the site of the patient presenting, these have yielded fairly consistent results, especially recently. If you look at the upper panel where it's done in the GP office in the United Kingdom, you see about 15% of the adults over age 65 who present with respiratory symptoms are PCR positive for RSV, in contrast to about 28% for flu. Similarly, at the Marshfield Clinic in Wisconsin over a 12-year period, where they were using the ILI definition of respiratory disease, because this was a study of influenza vaccine efficacy, and they simply looked for RSV in that same population. You see about the same numbers, 11% for RSV and flu, 23%. And then Kaiser Permanente, very, very similar kinds of numbers. As far as hospitalizations go, I just showed this slide to highlight a couple of things that we do learn from hospitalization studies, and that is the percentage of patients with flu and RSV in contrast, as a contrast, with underlying risk conditions that are identified. And I highlighted 3 of them here because they are the most important. That is congestive failure, COPD, and asthma. And I think it's important to think about these things when we talk about how we're going to implement our vaccines and who would benefit most. It's interesting to look at these in comparison to flu because it is interesting that RSV has higher numbers. And this is actually consistent with other studies. The same study was able to also show that over time, the mortality rate after discharge was somewhat higher or survival rate lower over a 12-month period than influenza. And I think this is an interesting study, but don't misread it, or at least I don't think we should misread it. And this is the data from the IV network of 20 states looking at what I would consider the post-pandemic or moving into the endemic period of COVID. And this is 3 viruses. Oops, sorry about that. Use the pointer. 3 viruses. You've got COVID here, which is the big beast in the slide here. And then you've got flu, which was fairly low, but we had an outbreak here. And then RSV as it dribbles along, and then we have a smaller outbreak here. But at least they were able to collect enough data on these study subjects to compare the outcome in the hospital between the 3 viruses, so they could compare RSV to flu, COVID to RSV. And in looking at each of these parameters of disease severity, oxygen needs, high flow oxygen needs, non-invasive ventilation, and ICU admission, and death, what you see is that they're all very similar, although RSV was in fact more likely to result in the need for this care and a higher in-house mortality rate. One of the reasons I think we have to be careful about interpreting this data is it's quite likely these individuals with RSV had higher number of comorbidities that are seriously going to affect their outcome of these respiratory infections. And so you have to take that into account. So I just want to highlight this. I know we discussed this yesterday, but I think it is important. How much disease are we missing, and how do we use that information going forward? So this is the study that was shown the other day, and I think just to go into a little bit of detail here, the Venn diagram speaks for itself, and what it shows is if you use different tests, how many extra people you will pick up that have RSV. So just going through the bar graph here, what it shows, the reference is a nasal swab, which is what's standardly used in all of our, or generally speaking, this is what's used in all of our adult studies as a standard. If you add sputum in those patients who can produce it, you get about a 20% or so increase in yield. If you add serology, you'll get up a bit higher, so you have a nasal swab and serology. And you can see as you add specimens, you do a little better. And this is, in highlighting here, shows about a 66% increase in diagnostic yield if you have a sputum, a saliva, and a nasal swab. And you, at least in a research setting, can actually take all three specimens and test them simultaneously. And if you add serology, you can even increase that to the point where you almost double the number of cases. And as you saw yesterday, some people have looked at the, how you might use this multiplier effect. But that's all theoretical, because what we really need to do is actually do this in real time in more studies and see how this changes some of the data that we collect. So who's at risk? And then, again, focusing on, will they benefit from vaccination? Clearly older age will, and this is data from the UK. Douglas Fleming, who's been at this, in this field for, actually predates my entrance into the field and Ann Falsey's entrance into the field of adult RSV infection. But the upper graph clearly shows that you have a hospitalization rate that is substantially related to age. And this is the older age group, above age 75. Here is a less old group, 65 to 74, and you can see there's quite a gap here. And down here in the red, you have 50 to 64, so the numbers are quite lower. And then you have, at the bottom, 18 to 49. But again, this should not be it could be somewhat misleading and make you make a decision that, well, this is the group, 75 and up, let's go after that. It's the most important group. And, based on age, it would certainly appear to be. But you also have to take into account underlying conditions. And this is data from Rochester that Angela Branch led. And I just want to, again, highlight those risk factors that increase your population-based incidence numbers as underlying conditions to account for hospitalization numbers. And you can see COPD is by far one of the more powerful factors, as well as congestive heart failure. But also, although not statistically significant, obesity was also somewhat greater, or it increased the population-based incidence of hospitalization, as did diabetes and asthma. This is some data that we generated from, this is a group of what is the incidence of long-term care facility residents' hospitalization rates, and what we find is it's about 500 per 100,000. This is a group that really hasn't been studied very carefully. So what other work do we have to do? Well, this is the demographics of one study by Begley that was recently published, and I want to just highlight, we've got the younger adult population that we shouldn't completely ignore, although they are not included in any of the vaccine recommendations. This is people ages, well, not completely, but 18 to 64 in this bracketed highlight, yet they're not uncommonly found in the hospital. And if you look at the prevalence of comorbid conditions, you also see some very interesting differences in terms of the prevalence of comorbidities according to age. And this graph shows this very nicely. And you can see that asthma is much more likely to be a comorbid condition in the younger group, and the same with obesity. But when you move to older adults, it's coronary disease, dementia, CVA. And then some of the other risk factors, such as congestive failure, is really not affected so much by age as a risk factor. And I just want to finish up with some social and economic determinants of RSV infection in adults. And this is important. This is the data from CDC looking at the median age of hospitalization in those over age 60 with RSV. And what you'll see here is that the non-Hispanic black group in the middle is significantly younger than the group as a whole. And I think this is important, and this was discussed in terms of the vaccine recommendations. And what this shows here, this is data from Yale, and it shows that socioeconomic effect of hospitalization rates in the adult population. And this is again data from our study in Rochester, and this also shows a marked difference in in age group according to race. So if you look at the white population compared to the black population, you see a marked difference in the ages of these hospitalized individuals. And the bottom line is that a very high percentage of minority populations as well as lower socioeconomic groups are hospitalized below the age 60. And so in summary, I'd just like to say there's a large but somewhat underestimated burden of disease in the populations that we're talking about. And we need to be a little more precise in how we measure them in the future. And some of the risk factors we've talked about are listed here. And we really need more data on socioeconomic factors. We need to study the under 60 group a little more carefully because they too may benefit from an RSV vaccine. And as I point out, we need to see how this data can inform our use of the vaccines we now have available. Thank you.
Speak about RSV transmissions, and I think this is early times to start thinking about whether RSV preventive strategies will reduce transmission, but I'll try to show data that we have on transmission that can inform us on our thinking around these effects of interventions. So these are my disclosures, and I have no conflict of interest to declare. To start, I would say from this particular graph, and as you've seen from other presenters, that RSV causes seasonal epidemics, and in this particular case, this is data from pediatric surveillance, long-term surveillance in Kilifi, at the top and at the bottom, Siaya in Western Kenya. And we see the associated annual epidemics that are seen to occur. And based on this particular data, it's clear that when RSV is introduced in a population that has susceptible pools, then the population gains immunity as people are infected, and at a certain point at the peak of the epidemic, you reach a certain threshold of population immunity that results to reduced transmission. I think the holy grail with immunization is to try to push people from being susceptible to being protected safely without causing illness, and that essentially leads to achieving that herd immunity threshold that will protect those people who are immunized and also those who are not in the population, and I think interfere with transmission. So the other point that I want to get from this particular slide is to show that in the tropics and in Kenya, we see a synchrony in terms of circulation of the virus. There's subnational variations in the occurrence of RSV, and this clearly indicates that if you have any interventions, the timing of delivery of this vaccination will be very critical and will be very challenging in situations where you have limited surveillance. So I will attempt to speak about the impact of the current interventions in infants and adults, and I will speak more on infants, on the possible reduction of transmission. And monoclonal antibodies and passive transfer of antibodies that is acquired through maternal vaccination is likely to be short-lived, and I'd be cautious in saying that because these are long-acting monoclonal antibodies, but the protection that they are likely to give is for a limited time. So as I said, it's the timing of delivery of these interventions to move people from being susceptible to protected would be very important. And again, we need to understand whether that has potential to reduce transmission. But taking a step back, looking at RSV, we know that it infects people throughout life. And in fact, actually, we really don't know whether natural infections prevents transmission to a great extent. So there's some bit of unknown in the dynamics or the correlates of protections that are acquired through natural infection. Findings from the Kilifi birth cohort by Eric Ouma showed that primary infections actually reduce the risk of reinfection for up to 6 months. So if we speculate a little bit, I would say that if vaccinations does not protect against infection, does not alter transmission, then the only major benefit that will arise from vaccination or interventions would be direct protection to those who receive it. So I think that is a sobering thought for us to have. But I will use this data from the household study that we conducted more than a decade ago to look at some of the parameters that will help us understand whether the current interventions will have an effect on transmission. And this is a study that was done in one administrative area in Kilifi in coastal Kenya, where we recruited for 7 households prior to the start of the RSV epidemic. These households had close to 500 participants and followed them closely by collecting nasopharyngeal swabs every 3 to 4 days for a period of 6 months during the RSV season. And RSV was detected through PCR, and we were able to distinguish between RSV group A and B. So one finding that we observed is that once RSV is introduced in the households, there is high transmission with high attack rates within the households. And overall, in the study, 40 out of 47 households had an RSV introduced in the family, and close to 80% of these introductions led to transmissions in the household. Taking that by itself, I would argue and say that preventing RSV from getting into the households is likely to reduce community spread of RSV in the population dramatically. And again, I think we've seen it from data that we're seeing that COVID mitigation measures, social distancing, masking, and hygiene are the effects it had on circulation of the virus. But from the data, one thing that we can do is to look at who introduces these infections to the households and target that for prevention. And analysis of the epilinks of transmissions within the households, especially to the primary cases that we looked at, the infants, we see that most of the index cases were older children between 2 and 12 years who are school-going. And only a few of the introductions were from mothers, 3.5%. And this particular evidence was sustained by analyzing whole genome data from the households, looking at the transmission chains using phylogenetic analysis, and also integrating that data in a model, in a mathematical model to look at who infects who within the households. And a more recent study in South Africa by Shelley Cohen, looking at multiple seasons in households in both rural and urban setting, also replicated the same findings. 60% of their index cases were from children who are below 12 years. And this is not new because even family studies that were done in the US by Caroline Brissol also identified older children as key to transmission. So I would argue then school-going child is important in transmission of RSV and preventing infection from getting into the household might require vaccinating or targeting this particular age group. The third point I want to make from the data that we have is that we saw that people who are symptomatic, index cases who are symptomatic were more likely to transmit the infection within the households as compared to those who did not have acute respiratory disease. And this taken together with the observation that those who are shedding for longer or had higher viral load are more likely to transmit, it would indicate that the mechanisms of transmission really requires having these symptoms. So sneezing, coughing, runny nose are important in transmission of infection. So interventions that are likely to reduce infectiousness, that is symptoms that participants have, or the duration of shedding are likely to provide some direct protection to the population. However, this is a far-fetched thought, and this will need also higher vaccination coverage and longer interventions with longer protections for us to achieve this herd immunity threshold. So in summary, I think I've shown that older children are important drivers for RSV transmission in the population, and interventions that are aimed to reduce transmission would likely need to target this particular age group. There have been several mathematical modeling studies that have shown the optimal age for vaccination is between 5 to 10 months to realize the greatest impact in terms of protecting people indirectly, especially the young infants. Another study utilizing data from the household showed cocooning method might also reduce transmission in the population, so that is vaccinating elder siblings of households with young infants just before the RSV season starts might interrupt transmissions. We can also explore school-based vaccination. But however, the utility or the impact of these alternate groups or methods of delivery of the vaccine would need predictive modeling to evaluate this impact, the impact of these, and especially in the context of the new interventions, the maternal vaccines, monoclonal antibodies, and also vaccines in the elderly population. So these kind of mathematical models will try and show whether a combination of these interventions will lead to moving people from being susceptible to protected up to a threshold where we can have reduced transmissions in the population. I'll end by thanking my former colleagues at Cambridge Wellcome Trust, current ones at CDC, and also recognize all the collaborations from University of Warwick, London School, and Cambridge, as well as the funding that we got from Wellcome Trust to conduct this household study. Thank you.
Thank you, Patrick, for the wonderful presentation. You want to come over and take a seat. So we now have some time for questions and also for a panel discussion if there's still.
Have now is a session focused on patient involvement in ethics, and we see that as an important part of the conversation we are having in Mumbai this year. The idea being that we need to talk about how we can get patient involvement center staged in our work, in our research, in our advocacy, and use that to further the field, and also speak about what are the kind of key ethical issues we all have to deal with and how do you respond to those. So to enable us to do that, we have a panel of speakers who will be covering the topics which include looking at a sort of broad-based perspective in global health around patient involvement and ethics, then doing a deep dive around RSV specifically and the kind of ethical issues which arise, and then two speakers who will be focusing more around the issue of patient involvement and how can we get patient representatives as well as caregivers involved in conversations around the field. And towards the end, we will have time for open discussion where we invite you to also share your comments, share your thoughts, ask questions of the panel, and hopefully that will help us have a more wholesome picture of how patient involvement and ethics can be actualized, can be implemented, and have some key takeaways at the end of this session. I also welcome my co-chair, Louis, to help steer this conversation, and look forward to the session. Thank you.
Yes, thank you very much. I think you've all noticed, and you were informed, that Barney Graham, who was supposed to chair this session because he is passionate about the topic, was supposed to be here. Unfortunately, for you guys, I have to replace him. And it's then my pleasure to introduce the first speaker, my co-chair, Dr. Anand Ban. He is a physician with a master's degree in bioethics from the University of Toronto. He's a researcher in global health, bioethics, mental health, and health policy. He's also adjunct professor at Jinopoya University here in India and past president of the International Associations of Bioethics. His work is focused on the ethics and equity in health, mental health, digital health, public health ethics, research ethics, community engagement, ethics of innovative technologies and ethics, training for professionals. So it's all about ethics. Anand is based in Bhopal, India, where he's also mentor and principal investigator on multiple grants with Sanat, a civil society organization. Anand is a member of the World Health Organization COVID-19 Ethics and Governance Working Group, the steering committee of the Global Forum of Bioethics in Research, and was part of the CIOM's working group on good governance practice for research institutions, which was recently published in a guidance document. Anand, you're going to set the stage. I look forward to your talk.
So as Louis said, the idea for me was to speak on patient involvement and ethics broadly. And that's why I titled my talk, Why Even a N of 1, which is of course something that we are all familiar with in the research sphere, why that matters and where it is coming from matters too. So I do have no relevant conflicts of interest to declare. A couple of weeks ago, the last issue of Lancet in January spoke to an important issue. This was focused on clinical oncology, but I think what is relevant here is a statement which was made, which is that we can have all kinds of endpoints, all kinds of issues of interest when we do research. But for patients, what really matters are primary endpoints around survival and quality of life improvement. And I would argue that that is a learning which can be also applied to any other field of global health, including the field of RSV. So how do we ensure that what we are doing actually makes difference on the ground, especially in the lives of patients who are at the center of what we want to do? Now, if you look at ethics broadly, I'm sure all of you know these principles. It's part of your 101 to ethics when you join institutions, when you do your research. So you're all familiar with the importance of focusing on autonomy through consent, ensuring confidentiality, rights, data use of samples, etc., being focused on how to deal with vulnerable populations, how do you deal with issues around benefits and non-maleficence, both direct and indirect, the risks or harms which could be potentially there, conflicts of interest issues, which is also very relevant because there can be interest in the products from, let's say, commercial angles, which could also influence the research and research outcomes. Also importantly, issues around justice. How do you select your participants? What kind of participants might be vulnerable? How do you address issues around stigma, discrimination, potential coercion to participate, equitable distribution of risks, benefits, as well as opportunities to participate, etc. So of course, this is standard fare for discussion around ethics. But I think what is relevant to today's discussion is to take some of this conversation from this very elite venue for the conference into the real world, where RSV plays a major havoc as part of an important public health problem, and that is the reality of many parts of low and middle income countries, including our own home country, India, where access to healthcare is a key ethics and equity issue. And I would tend to argue that that is the kind of ethical issues that we need to focus on from both a justice perspective, but also an equity perspective. How do we ensure that there is access to quality health, including new efficacious medical products and interventions, many of which we are speaking to through the entire conference this year as well, that the vaccines that we talk about, the antibodies that we talk about, how do we ensure that those reach these populations in remote parts which have been struggling to get quality health care for a long time. And also remembering that there is a larger picture here, which are the broader social determinants, including issues around poverty, malnutrition, poor education, unhealthy living conditions, and lack of access to healthcare, which are important factors to think about, right? So today is Valentine's Day. Many of you have had to travel to the conference during this week. And for an academic Valentine, of course, you need evidence. And so where is the evidence coming from is the next theme of what I want to talk about. A lot of challenge that we see is that research which happens in global health is largely still produced by high-income country institutions and researchers. Even the research which is happening purportedly in low and middle income countries is often led by researchers in high-income countries. And that, I think, is an important equity and ethics issue which we need to talk about. Of course, the field is also dominated often by older white male investigators. Things are changing. We are having certainly more diversity come through. But that is true also of LMIC contexts where older men who are in senior positions often dominate the discourse. And we need to change that as part of the reforms that we speak about. And especially in the context of RSV, we'll all remember the thalidomide tragedy as a good example of where overprotections in the name of protecting vulnerable populations and restrictions around that could sometimes also have negative downstream implications. And the lessons from that in terms of promoting more inclusion in clinical studies, in terms of the data that we get from entire population data sets, including among pregnant women, lactating women, and the focus being more on inclusion rather than exclusion becomes an important part. Now, transitioning that conversation to why patient involvement is important, I trained as a physician a few years ago. In a conference like this, I underwent a foot injury, and that foot injury led to, unfortunately, a situation where I had to get operated. When I was admitted, getting operated, and in the post-op period, for me, there was an interesting transformational experience of being on the other side. Being a physician who's been involved in clinical care versus being a patient who's receiving care, and being very cognizant of the kind of power inequities that you see between doctors and patients, providers and patients, of how you are often talked down to, how certain procedures like consent are mere formalities, makes you realize that patient involvement is a crucial part of bringing about change. And so this central tenet which comes from the disability field, which is nothing about us without us, would be the one takeaway message I would leave you with. If I had just an elevator pitch conversation with you, which was for 10 seconds, this would be the only thing I would say. That remember this as a takeaway from the session, that nothing about us without us should be a central focus area of the work that we do, whether you be a clinician, a researcher, an advocate, or a policymaker. And this, I think, has been also more exemplified by the experience of long COVID. Much before academics probably started engaging with it, it was patients who were experiencing these symptoms which were not really being talked about by their clinicians, not something which was there in the literature, who started seeking each other out, speaking to each other, and realizing that there was something interesting happening which they were dealing with, which they were struggling with, which led to long COVID being recognized. And then, of course, now over time, there are more funding opportunities, research, as well as involvement of persons with long COVID in the kind of response which is happening. And this also has meant that even bodies like WHO have started recognizing the need for developing frameworks, developing pathways, protocols to engage meaningfully, right? Because one way of engaging is a very tokenistic approach where you have someone just because you want to tick a box versus actually a meaningful engagement, and that is quite different. And this is extremely important because the history of medicine, the history of research has had a lot of mistrust because of historical harms which have happened, whether that be the Tuskegee syphilis study, whether that be the development of many interventions where communities were unethically treated, they were exploited, and this has led to a mistrust which still exists. I mean, if you see data from COVID from the US, you will see that many Afro-American communities had much higher morbidity and mortality because they would report much later to the institution only when they had serious symptoms, because they had a fundamental mistrust of the health system which persists, and that can, of course, have implications in many ways. And this is something that we've seen also in qualitative data which has been collected. We've been working in our research group on understanding vaccine access during COVID among marginalized communities, specific queer trans communities and persons with disability. And this is a thread which keeps coming through, that communities have often a lack of access to information, which makes it difficult for them to be able to understand why they should be engaging with the health system, why they should be undergoing an intervention. They have a hesitation because they don't understand the risk. No one is telling them that, giving them that information. No one is engaging with them in a more respectful manner. And of course, there's a lot of misinformation which is floating around. So unless we deal with some of these issues, any new interventions that we speak about in any field will be difficult to implement. So again, I think important to remember what needs to be done to really work with communities in an open manner by building trust, engaging early, listening to them, learning from them, getting to know who our target audience, who our participants are, how can we work in co-production of knowledge with them, what are the right communication methods given the context, given the audience, how do you ensure that consent is taken seriously, and communication not just being about the research study, but also the more downstream implications. And towards this, we've been doing in our research group, again, a lot of, we've been trying different models of co-production of knowledge, of working with relatively marginalized communities in developing models for development of knowledge, sharing of knowledge, photo voice stories as an example, doing advocacy meetings where community members are center stage, their stories are talked about, they are invited to be on panels, etc. And that has provided a model or a framework for us to be able to approach how to work respectfully with the area of lived experience. And this also, I think, is relevant to health providers because all of us are talking about stories of, the evidence from surveillance studies, the burden, etc. But at the front line, it's the health providers who struggle with that, who have to deal with the child struggling or the infant struggling to breathe when they have a serious infection. And we have to recognize their lived experience as well, whether those be frontline health providers or those be nurses or medical officers. And that's where I think working with these frontline health providers so that they are able to counsel, engage with patients and families becomes important. And hence I would urge us to think about avoiding, how to avoid tokenism, how do you engage in a respectful manner, how do you demonstrate respect for lived experience, how do you create spaces where people feel that they can be engaged, and how do we invest in creating capacity among patients and patient representatives and caregivers. And towards that, there is guidance now coming through. The Wellcome Trust has some really useful guidance which you can look up on their website, which might be relevant to all of you. And I will end by acknowledging that I was very happy as a first-time attendee at an RSV conference to see that there is content on the website, that there is a proactive effort to engage with patients, caregivers, to put out information. But certainly I would also urge us as a community to do more in this area. And hopefully today's conversation will also be a step in that direction. Thank you for your attention.
Thank you very much, Anand, for this very interesting start of a hopefully more interesting session to come. We hold the questions for the general discussion later on. It's now my pleasure to introduce Nathalie Mazur. We have known each other for many years. I asked Anand to introduce her because we know each other so well, but for practical reasons we said, okay, I can do it for once. We have been working for 10 years together. Nathalie is now a resident in pediatrics at the University Medical Center Utrecht. She is building her own research group and she is passionate about equity in RSV research and vaccine development. Nathalie, the floor is yours. Not yet.
Oh.
And now?
Good. Well, I'm really excited to speak today, and I think you probably feel the same excitement as I do, and that's mainly because now we really have the tools in hand to be able to prevent RSV, and that's what I want to discuss today. I want to propose a vision I have on vaccine equity, but more than anything, I would like to provoke a discussion to see really how you feel your role is in achieving vaccine equity, or do you have a different vision for the future of RSV? This is my disclosure slide. And I would like to start with why I stuck around so long doing RSV research, as Louis said. And that really comes down to patient impact. And some of you may have seen this video from a few years ago of Simone van Wijk, who was really brave in sharing her story of losing her 3-month-old son, Le Mondre, to RSV. And this really is the reason that I do this research. And she said to me when we were in her house, maybe if I had recognized the signs of respiratory distress, maybe if I had gone to the hospital earlier, maybe he would have still been alive. And those are really the words that I kind of keep with me every day, and why I get up in the morning and keep doing this research. But the important thing I would like to say is that now we have the tools to prevent RSV. So you could say, well, maybe if he had gotten RSV vaccine, Le Mondre would still be alive. But what I mainly want to do with you is look towards the future. So how can we ensure that vaccines get to the right place? And I think we set the right intentions 10 years ago. So the WHO convened and actually wrote a report saying we won't condone a scenario where a vaccine only becomes available in high-income markets. And I think it's interesting to reflect on the fact that 10 years ago we were saying this mainly because we didn't really get it right the first time. So as you may know, palivizumab has been market approved for 20 years, but due to high costs was not registered in a single low middle income country and could have had potentially a very high impact on infant death. So now I think, again, we've come to a very important moment in time where there's really an urgent dilemma, which I have called the RSV vaccine gap or potential gap. This morning we heard a lot about life-threatening RSV disease, and we heard that the burden of disease is inequitably distributed. So more than 97% of life-threatening disease occurs in low-middle income countries. And we're also at a time point where the vaccine landscape has really grown its fruits. So we have two potential interventions against infant mortality. And importantly, we're now at a very important moment where there's the potential risk of severe drug lag. And the picture on the left bottom, you can't see it very well, but that's a picture from the HIB introduction campaign from Gavi, where there was a drug lag of at least 11 years between introduction in Canada and introduction in the first lower middle income country. So this is something I think to me would be unacceptable if this happens again. And I know many people are working very hard to have it not be that way. And to put it very simply, there's a kind of painful misalignment between the burden of RSV on the left, which largely occurs in lower middle income countries, and potential vaccine access, which if you look at least historically is more or less guaranteed in high income countries. And we already know that there is deployment in high income countries, but if we don't take action, there might be a severe lag or even no access. So what I would like to advocate is really a vaccine equity, and that means that you would align vaccine access with burden and prioritize access for LMICs. So I don't mean don't introduce vaccines in high income countries. I think that will happen by itself. I'm not worried about that. But I think really focus our efforts on getting vaccines to where they are most needed and most importantly, where they'll have the highest impact. And this really calls for doing things differently in a system where we really need a novel drug development paradigm to achieve vaccine equity. And that means, to me, redefining success. So we may, and that's also what I hope to discuss later in the discussion with you and hear your perspective, but we may think, well, we're more or less there because we have a vaccine. But I would like to say success should be defined based on impact or lives saved. So really a new metric for success driven by impact. And a lot of the ethical debate around vaccine access has been sharpened recently due to the COVID pandemic, and I think we can also get some lessons learned from the pandemic. So on the left, you see vaccine access in the first year of the pandemic, which was really largely in high-income countries. And there was really an outcry from the WHO saying that really criticizing a me-first approach of vaccine access, where high-income countries massively bought vaccines and there was not a sufficient production for lower-income countries. And this was really coined as vaccine coloniality. So vaccine access was determined by market forces, but not determined by public health reasons. And one of the main ethical principles that was raised was the idea of reparative justice, so that we need to actually remediate what has happened in the past in order to move towards equity. And that means considering vaccines as a public good, but it also means doing things differently in clinical development. And the RSV vaccine development landscape has had a very similar approach, where nearly all candidates actually had a richest first approach, as I called it, but were really in development for a high-income country target. And there were actually only one or two with a lower middle income country target. And I would like to advocate to be able to achieve vaccine equity, that we need what I called a poorest first approach. Really advocate for children who are most vulnerable, and that I think we can do that in three ways. So the first is drug efficiency and question-based clinical development. And the second is a fail-fast approach to reduce wasted time and capital in clinical development. So that can be done by things such as controlled human infection models. And the third is really to have choices in drug development be guided by impact instead of profit. So it doesn't mean something cannot be profitable, but it means it is not the driving force. And that may call for alternative funding in the pharmaceutical industry. And I would like to highlight a few things we're doing at UMCU, which are really dedicated to a poorest first approach, because that's really where all our efforts are going. The first is the clinical development program of an intranasal palivizumab against RSV in preterm infants. In a phase 2b trial, we unfortunately found that it was not effective, and we had initiated the clinical development program for an affordable and acceptable prophylaxis. And in parallel, we've done a controlled human infection model, a phase 2 trial, of which we soon expect to be able to share the results. And the second I won't talk too much about, because I hope you'll all be at the Young Researchers Tournament, where Jonne will share her work on a new low-tech, low-cost device which can be used in lower middle income countries to measure functional antibodies against RSV in dried blood. Fail-fast approach has really been the hallmark of our research, where we've carried out the first outpatient RSV controlled human infection model this last year, and are developing a framework for outpatient studies which are less expensive and quicker in vaccine development. The Dutch government has funded InfectNL, which will be a hub for controlled human infection models and early development, and have its own GMP production facility which can be scaled up in times of a pandemic. And finally, a large project to de-risk the development of monoclonal antibodies in LMIC settings by looking at MARMs of global contemporary strains. And we're also focusing on understanding how we can best use vaccines for impact through the RSV Gold 3 network, where we found an expected high impact of a monoclonal or maternal vaccine in lower middle income country settings in 10 Gavi eligible countries. And the Gold Health Economic Study, where Nele and Farina are also in the audience who can talk more about that, for which we soon expect results of cost of illness and cost effectiveness analysis in lower middle income countries. And I would really like to commend the efforts of Gates MRI in the RSMO1 clinical development program, because it's really the only candidate with a primarily LMIC target and where affordability is really in the driving seat of clinical development. I've prepared a few slides, but this is actually more for the discussion on what I see as the role of, for example, the Gates Foundation, Gavi, PATH, pharmaceutical industries, regulatory bodies in accelerating vaccine equity, as well as academia, patients, because you're all here today. I hope you'll also comment on these things, but I think it really starts with the individual, because beyond our job, we're also just all individuals, and if we can align on a shared vision for what it means to achieve success with an RSV vaccine, I think then we can go far together. So if you remember anything from this talk, I hope it's this graph here, because I hope this graph is not only RSV burden, but it will also be the graph of RSV vaccine access, and that we can avoid the RSV vaccine gap by redefining success in terms of life saved instead of profit. And I would like to thank, above anything, actually, all the patients that have participated in our research, the collaborators, and everyone at UMCU as well. Thank you.
Thanks, Natalie, for taking the lens of ethics and equity to the field of RSV, advocating for poorest communities first, and also giving us some food for thought in terms of the conversation moving forward. Now we will transition to a talk by Rachel. Rachel is a patient advocate. Rachel is a university lecturer with a BA honors in social policy and public sector management, and also has a professional graduate certification in education. Rachel is currently studying for a master's degree in education and is a fellow of the Higher Education Academy, seeking senior fellowship this year. She's also the UK representative for the RSVNet Patient Advisory Board, where she seeks to raise awareness of RSV. In 2010, Rachel lost her son Alexander to RSV when he was just 13 weeks old. Rachel is married, is a mother to 6 boys, and will speak to us on the importance of patient involvement. Thank you, Rachel.
Thank you very much. It's an honour and a privilege to be here, so thank you for having me. So this presentation will aim to give you some background, thoughts and considerations of the patient perspective of RSV. This includes that of the parent and carers of those impacted by the virus. So first, the background. I am here standing in front of you because 14 years ago, I was unwillingly catapulted into the world of RSV when my 13-week-old son, Alexander, contracted the virus and died. For those of you that don't know, Alexander was my third son of 4 boys and was born a happy, healthy baby. The week of his death, Alexander had experienced cold-like symptoms but remained alert, albeit a little grumpy. The day before his death, I took him to see the local doctor as I was concerned about his colour and the cough that he had developed. I was told that he would be fine and may have a little bit of bronchiolitis. That evening, everything changed. Alexander woke from a sleep screaming. The cry was not normal, and my first thoughts were that of meningitis. He sounded like he was really struggling for breath, and an ambulance was called. Once we were at the hospital, Alexander arrested. And for 2.5 very long hours, the doctors and their teams fought to keep him alive. But he was just too small, and the virus was just far too big. Since then, it has become a personal mission of mine to raise awareness of RSV. Although I am a lecturer in my professional life, talking to you about my personal experiences is far more challenging than presenting anything I have ever learnt in the academic world. It is also hard when talking about and reflecting on personal experience to not get drawn into areas that, although important to me, may not offer you as medical practitioners any useful insight or information. What can I, as a mere parent, offer those of you that have studied for years about a subject I only know through trauma of the loss of my young son? However, I was reminded of the importance of research, vaccine development, and why I try and raise awareness in November of last year, when my nephew Tobias, who was only 7 months old at the time, became seriously ill with RSV. The impact on families when they have experienced or witnessed RSV is significant. Yes, the child is or has been sick, but the trauma it can cause the family is something that also needs to be considered and addressed. Both my sister and I recognised our babies were ill, and neither of us were first-time parents. We had experience. We both took our babies to doctors, yet were told that our babies would be fine. Even though we both challenged those doctors in front of us, we were sent away with what we now know were extremely unwell children. Tobias was in hospital, being assessed, and was released just hours before he was rushed into hospital in an ambulance, unconscious and barely breathing. He was transferred to the high dependency unit, fighting for his life. The experience of his parents very much reflected my own, and I question, how can we be 14 years on from when my own son lost his battle with RSV and yet still be witnessing such horrendous incidents, such as those as you can see on the screen? Thankfully, and with the care of amazing medical professionals such as yourselves, Tobias is now a very happy, noisy, 9-month-old baby, as you can see here. But the feeling or experience of not being listened to can have serious consequences. A parent, a carer, knows that person. They notice those tiny nuances that means something is wrong. By disregarding the voice of the patient, or in a baby's case, the parent, can induce feelings of doubt in one's own ability to recognize when our loved one is seriously ill. These feelings of doubt are one I have experienced many a time, both when Alexander was ill, but since with my other children. What exacerbates this is the aftermath of RSV, such as the remaining cough, which I'm sure you're all quite aware of, and knowing that your child at some point is highly likely to contract another illness in the not too distant future, as this is what happens with young children and their undeveloped immune systems. It is true to say that this is exactly what happened to my sister, as Tobias has only just been released again from hospital after a stay of another 12 days, a consequence of the potential impacts of having a severe RSV infection. Tobias' lungs and body just couldn't manage on their own, and he ended up back on oxygen and other supportive equipment, along with antibiotics and inhalers. Although my own experience has been through witnessing young babies becoming ill, it is important that I acknowledge that the elderly are also at risk of contracting serious illness as a result of catching RSV, and the difficulties this can present for them and their families. In some of the personal stories used in the RSV Awareness Week in November last year, there are a number of accounts from elderly patients who voiced just how awful contracting RSV was and how ill it made them. The anxiety that surrounds families and patients can be intense. What are we meant to do when turned away by those that we seek reassurance from? Not only when we present with an unwell person, but once that child or elderly person is released back into our sole care from the hospitals. What guidance is there to truly support the families and the patient? It can be very daunting taking a person back home when you have just witnessed them fighting for their lives. Even the loss of the sound of the breathing machines, the oxygen, the lack of alarms going off, and the hum of a hospital ward can be a difficult transition for families. Going from a noisy environment with the comfort of medical staff nearby to the quiet of a home can, in itself, be a terrifying experience, one which is enhanced by the self-doubt you have now potentially developed. The question then remains, how do we, those of us at conferences such as this, help those people in need of support? Immunisation. The introduction of preventative injections are a good start. And I know that years and years of work have gone into these. But immunisation in themselves may cause anxieties amongst families and patients. It is potentially a big ask for people to receive another new preventative injection, having fairly recently engaged with the COVID-19 vaccines and the concern that these caused when they first arrived on the scene. Not always helped by the high media coverage, which depicted the preventative injections in a less than favourable light. The advantage that the distribution of the COVID vaccine had was that it was in a global pandemic, and everyone knew what they were trying to be protected from. The difficulty the RSV immunisations may encounter are that the many people I have spoken to still seem to have little or no knowledge at all of RSV, and if this is the case, how do we expect immunisation programmes to work if people do not understand what they are being protected against? Educating people is so important. If families can recognise the impact of RSV and understand how prevention is far better than a reaction, then the benefits of not only the preventative injections, but preventative measures, such as those seen by the COVID pandemic, may help us have a bigger impact on reducing RSV numbers. Families need to understand that sometimes distance is the kindest gift you can ever give your loved one. If you are ill, not hugging that cute little baby when you just have a cold may save their life. Sometimes caring is not sharing. Beyond education for families is the continued education for health professionals. It became very apparent to me 14 years ago that many midwives and local doctors in the United Kingdom do not have the same awareness of RSV as those that work in the hospitals. Those that see the endless amounts of people being admitted and needing supportive therapy to survive and hopefully recover. For doctors and nurses who deal with RSV every day, it is reminding yourselves that the patient or parent in front of you does not deal with RSV on a regular basis, and it is highly likely that this is their first encounter with it. The patient, the parents, and the wider family may be tired, scared, and desperate for reassurance and help from you, the experts. This is a tall order for anyone, regardless of your training. I see it in my own profession, both in teachers, but also in the police recruits that I educate. The very real potential of suffering from compassion fatigue. Hansen et al. Recognise this in their article from the Journal of Psychology and argue that not only can professionals, such as yourselves, encounter compassion fatigue, but also suffer from emotional exhaustion. Needless to say, this does not stop families needing you to be able to empathise and explain what may seem like the most basic of information. Making sure that knowledge is not assumed, but rather that there are safeguards in place that allow you, if needed, to step away, so that the continued support of patients and their families is possible. This picture, as you can see, is that of a teddy bear and a blue ribbon on it, stitched together with a little star. These ribbons were made by my friends, family, and local community for Alexander's funeral. Everyone that attended wore one of these ribbons, and my children still have one each, pinned to one of these bears, even though the eldest is now 18 years old. This bear always reminds me of the empathy rather than sympathy that those I knew around me showed at the time of Alexander's death. That little moment in time that they all decided to stand alongside me to help share that pain. It is something that I will always appreciate for the rest of my life. A bit like the families that may only meet you for a few hours. Your empathy and guidance will stay with that person, whatever the outcome. So going forwards, we need to work together. This sounds basic, but the gap between families and those with medical training can be really hard. Managing a family's expectations is by no means an easy feat, but it can at times feel like you are the one being left in the dark about how and why some decisions are being made about your loved one. Honest, accurate, and timely information is key, along with understanding and empathy. To a family, this is possibly one of, if not the worst time of their lives, and may believe that to you, the medical staff, it is just another day at work. Families need to feel involved in the decision-making process. It can be really conflicting seeing your loved one needing life-saving support to 24 hours later being ready to go home. This quick change in pace can itself be difficult when you are the parent or carer who's been living on a high state of alert through fear and worry. Far more time needs to be taken in reassuring people and explaining the processes so that they understand and feel informed enough to have the confidence in themselves. Little changes can make a huge difference. For me, the loss of Alexander changed the course of my life. Some may say for the better. I would, of course, much rather my son was walking alongside me and that I could have seen him grow up. If I can help in some small way in the fight against RSV, then the memory of Alexander will be that of saving lives, even if it wasn't in time for him. Thank you very much for listening.
Thank you, Rachel, for sharing your personal story of loss, but also your key takeaways for this audience, for clinicians, and for everyone working in the field of RSV. We're very grateful for your participation. Can I also request Lisa to come on stage? Lisa Van Wiesen is also a member of the RSVNet Patient Advisory Board. Lisa joined the Patient Advisory Board 2 years ago after her 2-month-old daughter got hospitalized with RSV. She has been a midwife for 8 years and was not aware of RSV and its implications in her practice till her daughter got RSV, and she felt that as a health professional, she can join the patient network and make a change in public awareness around RSV. So on the panel, of course, we now have Rachel, Lisa, Natalie, as well as myself and my co-chair as well, Louis. So we are open to questions, comments. While that is happening, can I invite Lisa for sharing some of your own thoughts around how patients, patient families, and caregivers can be involved in the RSV field? If you want to share some introductory remarks.
What was the last word you said? I'm sorry.
Can you share your thoughts around how patients and families and caregivers can be involved in the field of RSV? And if you have any thoughts around the work that you've been doing with the Patient Advisory Network and its importance.
Well, I think one of the most important things could be is just listen to us, to what we think about RSV, about our babies, what we see in them, but also listen to us and talk to us if they are admitted, for example. When my daughter was admitted, there was one nurse that came in every day and just talked to me for 10 minutes. She couldn't do anything, but it really meant a lot to me. And I think just sharing that with us can mean a lot. So that could be one of the things, I think.
Hi, Andrew Lee, formerly of Merck, soon to be joining RSVNet as a fellow. So thank you so much for sharing your story. My question is about patient advocacy. Patient advocacy groups and companies developing new treatments or preventions often can have aligned goals. They both want to raise awareness of disease, and they both want to make the development of new treatments or vaccines faster. My question is, do you see a model for companies or industry involvement with patient advocacy groups where they can remain independent from each other? Because I've also read some say that, patient advocacy groups should not accept money from companies. So what is the panel's thoughts on that?
Can you repeat the final bit? Sorry.
Yeah, sorry, I went on a little bit long. Some people say that patient advocacy groups should not accept money from companies that are developing new treatments. But I believe that there is a way that patient advocacy groups can work with companies that are interested in new treatments, for example, for RSV, while still remaining independent. And I was wondering if you had thoughts about that.
Wow, that's a loaded question. Okay, so I think potentially there is a point that, and we would hope, from my perspective, that people could work together. I suppose the problem is with companies is sometimes that patients may feel that they are doing something for another point and not just to help the patient. So sometimes I think patients, when they see companies promoting ideas, they may feel it's maybe for a financial gain. So it'd be trying to iron out those, I'm not saying they're accurate, but sometimes those perceptions of patients. So I think we would need to find ways of ironing those out and building trust between different groups for that to maybe work effectively. I don't know what the panel thinks, but...
I think above anything, so I agree, I don't think I'm going to answer what is the right model to use, but I think we should definitely use the patient perspective. So I think a lot can be learned there, and I think that's the sentiment that you're echoing. So I hope at least that communication occurs, and whether that's financially compensated or not, I think that's where it gets difficult. But I think there's definitely added value in incorporating the patient voice into, especially into research questions and the path forward. And if you take Stephen Covey, begin with the end in mind, a lot of vaccine development goes in the wrong direction for many years because we don't listen to the patient about what their needs are. Sometimes they're very simple things, but if you start with the end in mind, which is the patient in the end, then I think that's another way to fail fast or reduce wasted time and capital, which is what I was discussing earlier.
Yeah, I mean, this whole issue of conflict of interest is not unique to companies working only with patients, right, or patient groups. It's also true of researchers or for that matter, policy advocacy. What is important though is transparency, and that is usually where the problems arise, that if it is not clear who's funding what and for what purpose, then that can always raise questions. And not just in the RSV field, but also in the NCDs field, this has become an issue sometimes, where patient advocacy organizations sometimes seem to be running orchestrated campaigns which are in line with what other actors want them to say, rather than actually representing patient and patient caregiver voices. And that's where I think the problem is. So if we can ensure that those lines are clear, and the idea is to highlight patient perspectives and caregiver perspectives, it's not only about the products that the company has an interest in, but genuinely wanting to promote patient interest. And maybe if those lines were clearer and more transparency was there, then it would be an easier sell in terms of public perception. But thank you for your question. I would also invite other comments and questions. In the meantime, Natalie, you were talking about how we could also transfer some of the expertise and the knowledge production as well as the manufacturing capacity to low and middle income countries. Do you want to maybe expound on that? Because you highlighted that of course is also an equity issue. The fact that most of the development as well as the production is happening in high income countries, the costs can be higher, the products are at a cost point which is going to make it extremely difficult for this to be adopted into the public sector and LMICs. So do you see any solutions coming through? I think you spoke about one, which was the under $5 example, but do you have other thoughts around this area?
Yeah, well, more than anything, I hope the audience will participate in this discussion, because I think there are a lot of people sitting here with hopefully thoughts about the answer to a very complicated problem, because there are many actors and a shared responsibility, which in the end means I think no one is responsible. But I think one of the main potential solutions is in the area of IP and patent law. At least if you look at the example from COVID, where there was really a push to create local production hubs, including really well-equipped mRNA technology transfer to lower middle income countries. And so maybe we can piggyback on the local production hubs that have been identified or that are now in the process of capacity building for COVID, and also utilize those for RSV. But the big dilemma is really in the area of patent law and commercialization, and is that really what should prevent access or not? It's really, and that's again why I would like to encourage a discussion, but it's about what norms do we feel are acceptable together with the larger community. So is a vaccine a commercial commodity, or is it really a public health good? And I think patient advocacy, but also advocacy on behalf of all of us, can play a big role in changing that norm.
So we have a question from the audience. While it would be good to hear some of the representatives of companies who are here. I'm sure you have dilemmas. Don't worry, you don't have to give us solutions, but it would be nice to hear your dilemmas. In one ear you hear the voice of the people sitting next to me, and in your other ear you hear your company that wants to make profits, or in your head you understand you work for a commercial company, in your heart you feel you need to do the right thing. And it would be very interesting, and I'm sure rewarding for the people here, to hear these dilemmas, how you cope with them and what you think about them, even if you don't have direct solutions or agree with anything we have just said. It would be good to hear some of the companies people speak up. Go ahead.
Hi, my name is Sonia from Spain. And so first comment to the families. Years ago, before I even started working in research, my daughter was hospitalized in United States with a bronchiolitis, and she was in the ICU unit. And I remember being 7 days there, like 24 hours without putting a foot outside the hospitals, and I felt that feeling of how hard it is to have your baby in the hospital. And then all the sequela that came after. So when I started working in this, I did research in other things before. When I had the opportunity, I thought, okay, this study was meant for me because I had that issue before. So yeah, I think that all of the people doing the research, they also have the personal experience, either their own families or friends that had been through this disease before. So I think most of us probably completely empathize with you. And my question to Natalie, before I worked with RSV, I was working with neglected diseases, particularly with Leishmania. And there was one PhD student she was doing an analysis about public-private partnerships for drug development. I don't know if that is an option for RSV or is just an option for neglected diseases. Have you ever heard about public-private partnerships?
Yeah, I think that's a very good point. So that's something that's already happening in many collaborations in RSV. And I think if you look at what I mentioned earlier, if you want to move towards impact maximalization instead of profit-driven decisions, then where, for example, funding comes from is important, because if the funders demand a certain return on investment, then decisions will always be determined by profit. But if funders accept some form of profit, but the leading principle is, for example, impact, then decisions can be made differently. So I think there are many options also for public funding, but also for private funding, such as venture philanthropy or other options along those lines. I think the unique thing about RSV, so for neglected diseases, there is no high income market. So that makes it much more difficult. But for RSV, there is a high income market. So that makes things, in my eyes at least, much easier, because there will be a profit, but
that guaranteed profit can also be an opportunity for more equitable distribution. So on some level, doesn't necessarily come back to your question on public-private partnerships, but I think it's important to make that distinction because that distinction gives us more opportunities than the challenges with funding for neglected diseases.
Thank you, very good discussion. I'd like to give my comments, especially to give some examples in our area, like Meghalaya or any tribal area in India is known for very high prevalence of malnutrition and mortality. And most of the interventions by governments and other institutes were hospital-based. We had a lot of surveys and focus group discussions with the community, and we realized that community want community-based services through the local person and in the form of local food rather than the industrial food. So we did a randomized control trial where we involved the community since the planning, designing of the study. Even selection of workers was done with the help of the community. All the interventions were planned with the help of community, and we could significantly reduce the mortality, prevalence, as well as recovery rate was very good for malnutrition. So that was the advantage of community engagement. Second thing, regarding COVID vaccination, the acceptance of COVID vaccination was very low in our area, less than 5%. Initially we thought probably there are some miscommunication gap, patients are afraid of vaccines, but later on we realized that most of the people, they are working family, they are working going to field, and they can't afford to go to hospital which is 25 to 30 kilometers. Then we discussed with the community and realized that if we can provide the services in the community itself and during evening hour rather than during morning hours, then that might increase the chance of acceptance. And it happened. When we went to the villages with the government system and started vaccination during evening hours in the village, there was significant improvement in vaccination. So definitely involvement of the community members, the caregivers and relatives of patients will play a very important role for acceptance of this RSV vaccination. It's my suggestion. Thank you.
Could I just briefly reflect on that? I think that's great. So this is what I hoped, that people reflect on, kind of what can I do in my role to move towards vaccine equity. And I think community engagement and creating demand. So even if there's affordability, it doesn't ensure demand or acceptance. And this sounds like a ground up way to do that. So thank you for sharing that, Ashish.
Thanks. Yeah, thanks, Ashish. So, I mean, some of our work during COVID, we realized that it's very easy to label communities as being vaccine hesitant. You know, vaccine hesitancy as a word is thrown out very liberally, even in policy circles. But what doesn't usually happen is an examination of why is it that communities sometimes are hesitant about new interventions. And when you speak to them, you realize that there are larger systemic issues around health systems not being responsive to their health needs, health systems never reaching out to them. If you just have an experience where communities have not had healthcare access for decades, and then suddenly someone is coming to your door and saying, we are running an adult vaccination program and we want to give you shots for a particular disease, then they're going to wonder, where were you all these years and why is it that today suddenly you've remembered us, right? And so of course they are distrustful, they sometimes have questions, their information might not be shared with them. And when you do, and when you have honest conversations with them and you try to design your program to be responsive to their needs and their aspirations and help them understand what is it that you're bringing to the table, then of course you will start to see that trust starts building up and probably acceptance goes up, and hopefully that's the kind of change which is relevant in terms of any design of an intervention, whether that be a vaccine, antibody, whether that be a food or nutrition based intervention, etc. So again, thanks Ashish for bringing it up.
Hi, Iona Manjal. I'm a pediatrician, a parent, and I lead Pfizer's RSV clinical vaccine programs, and I'm very privileged to be driven in that order. I had patients who said, just fix this in their ultimate frustration. I'm a parent of a premature infant who had bronchiolitis in the first 3 months of life. And then I have the absolute privilege of being able to lead our RSV clinical programs, and you're asking what we wrestle with. And because our programs, and predominantly our maternal program, is driven towards our desire to reach LMICs, I think what we wrestle with is what our endpoint should be. Obviously, when we design our clinical trials, we know that the LMICs are going to rely on the regulatory pathways in high-income countries, so we want to appeal to that. But we really appreciate the work that you're doing, not only from the patient advocacy and ethics perspective, but to drive meaningful endpoints and to tell us. So I think that's what I wrestle with when we're designing our programs. We have RSV, which, as you note, is a high-income country desirable vaccine, but we also have GBS coming up, which is, a very, very predominantly, and so anything that you can do to reflect upon key drivers in LMICs, I really appreciate the reflection on mortality data and et cetera, and anything that we can do to make pronouncements which could really drive our endpoints and advocate for regulatory pathways for those endpoints to be important would be really great for us, I think, in industry.
Sure. I actually asked for you to come to the microphone, or as a representative, so I'll take this question. I think it is very fair to ask to help out with understanding endpoints in clinical trials in LMICs. So the point is taken. I think you should ask the people within the community of the LMIC. So I feel a little bit reluctant to talk on behalf of them, and I think mortality or death is not a good endpoint for obvious reasons. When you look at the current trial vaccine development for maternal RSV vaccination, so I'm just reflecting on RSV maternal vaccination, I think both at the efficacy and the safety endpoints, there are things in LMICs to address. I think many in the audience, but not everybody, understands that immunogenicity as well as antibody placental transfer may be different in LMIC. We need to understand this, and the only way to do that is to do an impact trial, as you and people within the Gates Foundation and others are developing or thinking about or planning. I think this is critically important. I'm not sure that correlates of protection are so different in LMIC. So I think if we can prevent some form of severe RSV, lower respiratory tract infection, I think it would be sufficiently good. I think we have learned from the Novavax trial. Some of you remember that we were very hopeful in one of the RSVVW meetings that that would be like a solution. It turned out to be that efficacy was too low. But in their trial development, in their vaccine development, they had a very complicated endpoint definition, and that hampered the trial. So I think if you go into LMIC, you need to be cognizant of the fact that it may be more difficult to get the perfect answer. I think it needs to be good, and you need to be able to ensure that you get it from trial participants. In terms of safety, I really don't know. I think there is some debate on premature birth in relation to maternal vaccination. My personal view is that we haven't seen anything more than some number imbalance, and I don't see any biological reason that that would be very different in an LMIC than in high-income countries. I haven't heard anybody say that he would understand that that would be different, but I think based on everything you heard, including some form of vaccine hesitancy that is specific to LMIC that happens in people with a different background, I think also safety should be carefully studied. And I would be very happy to hear others. I know Heather, I'm not sure whether you're here in the audience, oh, there you are. I remember that we have discussed that, sorry, you were sitting behind Joyce, that we have discussed this in the past. So both, and if you want to contribute, feel free, to safety and efficacy. I think involving patients from LMIC, there was a question about paying patients stay away from paying patients. Please stay away from paying them. You didn't hear any patient asking for money, so don't offer it. I don't think it's going to work. Just listen. That's I think, what should be done. And thank you for bringing this up. So this was exactly what I hoped you would talk about. Heather, I think you're going to add.
Yeah, thank you very much for asking for my thoughts on this, and I agree with everything that you've said. I think one issue with these trials is that there's poor representation from low middle income countries, and we hear this from WHO when the data are reviewed, and we hear this from countries about how can we approve something where we have, where most of the data comes from high income country settings and where low middle income countries may be represented by South Africa, for example. So I think it's a challenge to companies that these trials are often more difficult to do in those settings, and we understand that. But, I think doing trials with a greater low middle income country representation, but also including populations that may be particularly vulnerable to disease. So, for example, this has been mentioned before, the Pfizer trial didn't include HIV-infected women. It doesn't include any women who may be at high risk or higher risk of poorer birth outcomes. And one understands that in terms of the initial trial for a vaccine and where you're going to file in the FDA or EU initially. But it does raise issues in terms of the generalizability of results to low middle income country settings. And I think those are crucial issues for companies. And then, of course, cost is a key driver here. And that's the challenge, I suppose, to companies who have done incredible work in terms of developing effective preventive options. How are you going to make those affordable in low middle income country settings? Have you considered tiered pricing? Have you considered mechanisms to actually get those out to the children who need them, as has been highlighted very strongly today?
So we are almost at time, but I know two colleagues are still waiting. So if you could both ask your questions or share your comments, and then we'll get one last round from the panel.
Yes, exactly. As the conference organizer, I'm a bit in conflict with myself, saying, hey, it's time. But I just want to say to Rachel and Lisa, so thanks very much again to be here on behalf of our patient network. Thanks also for sharing your story. And Rachel, you talked about education. That is really important, and I know that we have talked with you too, both on how to involve parents on developing educational materials and to make sure awareness of the disease. Can you just maybe 1 minute or so, I don't know, both of you or Lisa, discuss on how do you see your role in education of the general public?
Can we get you maybe also to ask your question, sir?
Yeah, no, this is more just a commentary question. My name is Gustavo Dayana. I work at Sanofi. I am also the global clinical franchise lead for RSV vaccine. And I really find this very helpful, and I think this is a collaboration that we have to really put in place, because working on the vaccine on the private sector, you may have the idea that this is the dark side, or some people may have that, and this is just profit. Actually, I used to work for the government. I moved, they said, well, they always told me you are going to the dark side. And I found out that it's not. It's really, we really work for the patients, as we all do. We are physicians, we are pediatricians, and I think we all have the same goal. So this should be noted, and I think we are all doing the same thing together, so we should be collaborating. So that's one point. And then in terms of LMICs, I think we also want to include as much as we can countries that can be in the LMIC category, because we have idea on how the vaccine works in different environments. That's what we're trying to do, but we also have regulatory requirements to include some countries that are high-level countries, and we also have to do that. But I think the idea at Sanofi at least is to include LMICs as well. So thank you, and thank you for putting this together. I think it's very good. Thanks.
Thank you. Education, I mean, obviously I'm a lecturer by trade, so education is obviously important for me because I do it every day. But educating parents around RSV, I think, can be quite difficult. We have to be mindful of assumed knowledge because I think those of us that are now in the RSV world, we assume certain elements, even simple things like, can someone take a temperature? I know not everyone who has RSV has a temperature. Alexander had no temperature. But just simple things, we have to be mindful of that. I think that's the first point. Starting education early, when someone already has RSV, is kind of too late. Because I can only talk from a parent perspective, but never mind how educated you are. I know a lot about first aid, I've taught it. But you panic. If your child's in crisis and can't breathe, your logical brain, I don't want to talk for everyone, but mine definitely stopped working for a moment before I remembered that I needed to start resuscitation. Not everyone knows those things, so we need to start the education much, much earlier. So, and I know Lisa will expand on this, but even before babies are born, we need to be educating parents. You know, in the UK, we get given packs on various vaccinations they might encounter or various viruses. That person might not read it, but we need to be starting early, and we also need to do it again and again. We know from education that if you teach someone something once, they're unlikely to remember it for a long period of time. They'll only retain it for a small amount of time. So it's continual education, not just one time. So tricky. But yeah, I think if everyone's working together, that is an achievable thing, I hope.
I really agree that we can educate pregnant women and parents as well, indeed, because that's where it all starts. Those people are becoming parents, they are trying to learn everything about becoming a parent, about their babies, about who is going to take care of them after the baby is born, for example. So we can really start there by educating parents as well, and we can do that by talking to them about it, by giving booklets, by the bureaus that are going to look after them, have them explain it, for example, and then we can tell them again after this care is being taken over by the GP, the family doctor, whoever it's going to be. But if you do say it more often, it will stick to them, I think. And if you start with that in a pregnancy, when everything is very important, what you're going to learn for your new baby, I think it's going to stick, because people learn about children's diseases in a pregnancy already, and this should be one of them, and it's not. At least it's not in the Netherlands still, and I think for many countries it's still not part of that education. So we can really just start over there. And I have started it after my experience with my daughter. I started to talk about about RSV to people in a pregnancy and giving them advices on when to call their GP if their baby is short of breath or whatever. And for now it's only me. It's a small impact, but it is an impact, because I can see that people are calling us or the GP more often if they see that their baby has symptoms. So they are taking up on it, even if it's a small impact. So I think if we all do that more, with whomever is going to take care for people in a pregnancy, I think we can make a bigger impact as we go along.
Feel free to close. All right, so thank you everyone. Thank you for your attention. Thank you for attending the panel. Our thanks to all the panelists for sharing their perspectives. We will now take a break, and then we have the next session. So feel free to go for your tea or coffee break, and thank you again for attending.
Good afternoon. Please take your seat so we can resume the conference with this afternoon session dedicated to the Young Researchers Tournament and the display of the best science abstracts that have been submitted to this conference. I am Paolo Manzoni from Torino, Italy, a neonatologist and a founder member of RSVNet. I'm playing here the role of the senior assessor together with my junior colleague, Ashley Murray. I only want you to know some organizational and background details behind this session. We give a great value to this session. This has been traditional for RSVNet and for the RSVNet conference. And we had a great number of abstracts submitted to be evaluated. This year we had 51 submissions, and the process of selection was involving a number of assessors, namely myself and my colleague Ashley, with several criteria that have been used to quantify the quality and the merits of his single abstracts. At the end of this process, we came out with the 4 that you will be seeing displayed and commented today by the authors. We'll have 50 minutes for each presentation. Out of them, a maximum of 11 minutes will be dedicated to the presentation, and we'll have at least 3-4 minutes for questions and answers. Having said that, it's my pleasure to leave the microphone and the stage to the junior very expert member of this chairmanship, Ashley Murray. She's here because she's the winner in charge. Last year she won this tournament, and she so qualified to be on the other side of the stage, assessing the merits and conducting together with me this session. Please, Ashley, welcome, and the floor is yours.
Oh my goodness, thank you so much. Thank you to thank you. So I just want to first off say thank you to the RSVNet folks for inviting me to come back this year to do this job with Paolo and to announce who will be the winner for this year. So I'm very grateful for that. And I guess I'm to speak on behalf of what the experience means to young researchers to have participated in this competition, and what that means for, you know, the continued improvement of young researchers in the RSV field. So having been in the crowd last year, it was definitely a lot more nerve-wracking than being up here now this year. But I just want to say that, you know, coming to these type of events, it can be, you know, intimidating when you are in a room with people that have years or decades worth of experience in the field, and then getting the opportunity as someone who's early in their career to, you know, contribute what little they can with the research they've done up into that stage is extremely meaningful. And I think that this is one way that the organization continue to foster the importance of encouraging young researchers in the field. And, you know, this will be the next, you know, generation of people that in 20, 30, 40 years switch roles with the people that are up here today. So I just want to thank you for the RSVNet people for having this competition every year, and I'm honored to be a part of the crew that gets to serve in this role. I think that's all I have to say about that. Yeah.
So I think we can kick off the session, and so Ashley, please, it's your task to introduce the first one.
So it's my great pleasure to introduce the first speaker for today. Dr. Anubhava Rajan is now the assistant professor at the Indian Institute of Technology in Madras in India. She is the principal investigator of the Orga Ahmed Research Lab, and her research lab focuses on the development of personalized medicine and targeted drug therapies using organoid models. So it is my great pleasure to welcome Anu to the stage.
Thank you, Ashley, for the wonderful introduction, and thank you, everyone. And I would like to extend my sincere thanks to the RSVNet group for giving this wonderful opportunity to present my work here. So I'm going to be talking about decoding age-specific RSV tropism using single-cell RNA sequencing in human nose organoids. So I don't have any conflict of interest to declare, and we have 4 posters from our group, and they will be presented at the virology and immunology section. So when you have time, please take a look at them. We have been hearing a lot about severity of RSV infection in children, and what I'm going to tell you today a little bit differently is what contributes to this severity of infection at the cellular level. So to give an outline about my talk, first I'm going to talk about how age drives RSV tropism, and the next question is what are the cellular features, like are there differences in cell types between an adult and a pediatric patient that drives this RSV tropism. So for my study, we are using human nose organoids as a model system to study RSV infection. First, I would like to briefly introduce our model system because this is a previously published work. We have developed human nose organoids from nasal wash samples, and with these human nose organoids, we developed them into air-liquid interface culture system to study RSV infection. An important feature about human nose organoid is that because each of these organoids are derived from an individual, they retain the genetic background of the person, and one organoid represents one person essentially. So to briefly touch upon, here is some data on how RSV infects a human nose organoid cell. I just want to bring importance to 2 colors, which are the yellow here is RSV and the green is the presence of cilia. So these model systems have respiratory ciliary cells just like in our humans, and RSV nicely infects them. And a clinical hallmark feature of RSV is hypersecretion of mucus, and that is shown by these green cells here, where at the early infection there is less mucus and more of mucus production upon RSV infection. So with that information, I would like to go ahead with the 2 research important questions that we are asking. One, by obtaining human nose organoids from adult and pediatric samples, can we identify differences in RSV tropism between an adult and a pediatric patient? And what are the cell types that drives it? Mainly because clinically we know the relevance of age, and also ciliary cells are thought to be the cells that RSV infect. So is it only the ciliary cells that are being infected by RSV, or are there other cell types? To answer these questions, we use the approach of single-cell RNA sequencing over bulk RNA sequencing. And why is that? Because when we do bulk RNA sequencing, we do know that the essence of all the signal get masked upon with the bulk sample load, so we don't get the resolution that's happening at single cell level. So in order to identify what's happening at every single cell in the nasal epithelium, we perform single cell transcriptomic, and our approach was to use human nose organoids that were infected with RSV up until 5 days, and then we isolated every single cell from them using flow cytometry method, and then we subjected it to deep sequencing. So this is our working pipeline for our data output. And first I'm going to show you a snippet of the first set of data. This is adult human nose organoids. Here I call this a little bird. It looks like a bird to me. I don't know how it looks like to everyone. So what you see here is a reduction plot of over 10,000 individual cells that are present in the nasal epithelium. So these are color-coded based upon the cell types. For example, the blue represents the ciliary cells. There are multiple ciliary cells existing, and the reds are the different basal cell population, which are the stem cells of the epithelium that are present in the organoids, and the green are the secretory cells like goblet cells, club cells that produces mucus in the epithelium. So here what you will see is that there are 10 different cell clusters, meaning our nasal epithelium have 10 different cell type populations that are present. And in the pediatric HNOs, what you will notice first is there are more cell clusters. Unlike adult, there are more cell clusters, and what are they? They are mainly proliferative cells, meaning these cells are under more cycling condition in comparison or contrast to adult, and there are also more types of basal stem cell population demonstrating that they are in a transitional phase or a growth phase. So to just give a take-home message, pediatric HNOs display a lot more type of cell type, and they also have a distinct proliferative state in comparison to adults. The next thing is what cells are getting infected by RSV, right? So that's what I want to talk about. So in order to identify that, we used 5 different genes of RSV, which are N, P, NS1, NS2, and M2-1. Only if I see the presence of all the 5 genes in one cell, I call that an RSV positive. Otherwise, it's just a noise or error from the sequencing method. So with that as an idea, here I present data from 3 sample set, mock, RSV-A and RSV-B. And what you see here is mock, of course, you don't see any RSV. RSV is color-coded, A by blue color and B is by red. What you can see is mock, there is no A or B, but in RSV-A, you see that clustered in this particular population. And similarly and consistently, you see the same thing with RSV-B, and this is nothing but a ciliary cell. Oh, this is a little bit of unhappy news because we know ciliary cells are already infected. What was striking is in pediatric data set, here you have mock RSV-A and B, the colors are the same. You will see the presence of RSV in this population, which are ciliary cell, but you will also notice the presence of RSV here and over here, which are basal cells and ionocytes, which are unique cell type that we have identified in our study. I'll of course go over the details of it in the upcoming slide, but I want to move on and talk about what happens in the ciliary cell population, because ciliary cells are thought to be the most susceptible cells to be infected by RSV. So I showed you data on what happens globally with the entire epithelium. Now the next set of data that I'm going to show you is I'm taking just the ciliary cells out of the entire epithelium and I'm subtyping into different epithelia, in different ciliary cell types. So here in the adult Asian nose, I want to bring your attention to the bottom of the graph where I have labeled them as primary cilia, and deuterosomal cells are the precursors of ciliary cells, and then mucociliary cells are one that can produce mucus and also have a ciliary phenotype, and there are motile ciliary cells which are nothing but the cells that actually beat on the respiratory epithelium. So in the nasal epithelium, you don't just have one type of cilia, you have 4 types of ciliary cell, and that was a novel discovery for us itself. In the pediatric population, in addition to these cells, we also identified 2 other cell types, which are these boxes shown here, which are cycling ciliary cells, meaning these cells continue to replicate more and more in a very enriched and proliferative cell state, which makes them very distinct from what we see in adult. Now, where do we find RSV in the ciliary cell population? So that was our next question. So I showed you this graph earlier. Now I'm going to bring out the RSV genes that I was measuring. Here you can see that majority of the RSV gene for A as well as B is present only in this cell group, which is nothing but the red primary ciliary cells, but not in these tail groups, which are the motile ciliary cell. So it seems like RSV preferentially binds only to the primary ciliary cells and not the motile ciliary cells. Of course, it logically makes sense when something is continuing to beat, maybe RSV does not like to bind, or there are more mechanisms associated with it. And now I want to bring out the pediatric HNOs, and where do we find RSV here? Again, what we saw was this group that you see here, which are nothing but the primary ciliary cells. That's where we see the presence of RSV-A and again in RSV-B, but in less of your motile ciliary cells, showing and demonstrating again that primary ciliary cells are the target for RSV. The next set that I want to talk about are the basal cells and ionocytes, which I mentioned previously. First, I want to introduce to you or reintroduce you all to this graph where we see the presence of RSV, especially in the pediatric samples in the basal cell and the ionocyte population, not in the adult. And we subclustered the basal cells again, looking at what are the different types of basal cell, because generally we think basal cell as one stem cell population that gives rise to the pseudostratified epithelium. What we identified was there is like a multipotent basal cell, and there are basal cells that are primed to become secretory cell type, and there are also novel cells which I call them as activated basal cell, which is highly enriched in the infectious population. So here I want to bring highlight about RSV A and B, where you see in this trail, which are nothing but the activated basal cell, that's where RSV preferentially binds, unlike the other basal cell population. Once again, demonstrating a new tropism for RSV, and these activated basal cells are marked by the markers KRT23 and KRT17, unlike the multipotent basal cell, which was marked by KRT5. So, and in the ionocytes, we identified 3 different types of ionocytes, activated ionocytes in orange, and the tough cells which are a little bit present in the green. And we noticed that RSV preferentially infects the activated ionocytes. So to summarize, what I've shown you today is like the primary cilia is the main target for RSV within the ciliary cell population. And I've also identified basal cells and ionocytes are novel cell types for RSV to infect, and only in the pediatric human nose organoids, not the adults. Of course, the validation mechanism using immunohistochemistry and flow cytometry is currently in progress, and eventually we would like to identify the host receptor that drives this tropism. So with that, I would like to thank my mentors and the entire team for this awesome data and the funding sources for the study. I'm happy to take questions.
So if there are any questions for Anu, please feel free to come up. We have time for a few, but I can start with one. So for the activated basal cell population that you found, is it known what the lineage of that cell population goes on to become and what the function of those activated basal cells are?
Sure. So activated basal cells, so if she's asking about the lineage or where the activated basal cells are going or where it comes from, so it is hypothesized that all the basal different cell types start with the multipotent basal cell, but based upon the signaling cue, they go on to become different types of basal cell. These activated basal cells have a role to play majorly with repair and regenerative process, and especially in terms of an assault or injury that's happening.
We have time for one more.
I was just wondering, this is all organoids.
Yes.
Is there any way to translate this into the real life situation?
That's an excellent question. So how do we take this finding to the real life situation? One thing would be to look at maybe the pathology or the histology samples, if we can have access to the pediatric individuals who have severe infection with RSV and tag markers for these specific cell types in those slides. So that will be a way, but of course I don't know which parent will be willing to subject their child to it, or maybe if there are exceptional situations like bronchoscopy or a transplantation involved, and if we have access to the sample, why not look at it real life?
You could actually start in some of the animal models, of course, and the markers may not be exactly the same, I realize that.
Yeah. Animal models, of course, like, but I don't know if the markers for these cell types would be the same and if it would infect the same level. Yeah. Okay. Thank you.
Thank you very much. Thank you. Needless to say that all speakers remain available after the end of the session for further questions if you have not time to place it now. So for sake of time, we need to limit, and we go straight to the next speaker, Joanne Terstappen from the Wilhelmina Children's Hospital in Utrecht. She is a young doctor belonging and working with a group of research on RSV led by Louis Bont. Please, John, come to the stage.
All right, thank you. So indeed, I will be talking on behalf of my entire group about RSV neutralizing antibodies in dried blood. But we've already been talking, we're nearing the end of the day. We've been talking about science and RSV for 2 days. Really, really excellent, of course, as RSV researchers. We're quite happy about that. But I would really also like to take the opportunity to talk about something else that's really important to me, and that is the mountains. Everybody needs a place to really come to rest and to reflect on everything. For me, that is the mountains. And I'd like to take you through a recent tour that I did. It's a multi-day hike in Switzerland, where you have to ascend and descend 7 peaks in a couple of days, so really quite breathtaking. We were already 3 days in, so our last day ahead, and we had the option to make a choice. Life is all about choices, and we were on the peak of one of the mountains, and we could either go all the way down and all the way back up, but that would add a descent and an ascent of 800 meters. Or we could decide to take a little bit more of a daring cliff road where you really had to walk sort of side by side, foot by foot, deep drop on your left, but it would save our energy and resources. And so I was really going for, we should take that road, even if it is a bit scary. And I'll tell you at the end what my hiking partner thought of that. So why am I telling you this? Is this even really, really relevant? Well, I think it is, because we are constantly making choices. Also in vaccine trials, we need to decide, do we do A or B? Do we take a safe, more conventional route, or do we try something more innovative, even if it might be controversial? And so you could take it as simply as using dried blood rather than serum. The key correlate of protection in all of our vaccine trials is still neutralizing antibodies, and we take that from serum, which is, rather invasive to get, especially in children. If we're talking about infants, it's really difficult to obtain, even if you have a trained phlebotomist. You would need immediate processing, but the good thing from it, even though it costs more resources, if you're taking a venipuncture, you also have access to your immune cells. So that's the good thing that comes out of it. On the other hand, dried blood would require less resources. It's not as invasive because it's only a small finger prick, or in children maybe a heel prick. You don't need the cold chain, no immediate processing, and so it's really quite saving any resources or costs, and it's logistically just a lot easier, especially if you're planning to do remote or trials in remote areas and LMICs where the majority of the burden is. And so dried blood is used a lot, so why am I here really advertising it? It's used a lot, but only in concentration of antibodies, and we don't really know if your antibodies retain their function or their neutralization. So that's what we did. We did that together with the Gates Medical Research Institute while they were doing a phase 1 trial with their antibody called RSVVW'. This is an affordable antibody aimed at the pre-F site 0. Which really is targeted for low-middle income countries. And what was nice about it is that we were allowed to do a head-to-head comparison. So at 3 time points pre-dose, 3 months and 5 months, they took both the capillary dried blood as well as serum. So we could compare those samples. And we did that on a neutralization assay that we got from Barney Graham's group from the NIH, where indeed we have these 2 samples. We elute the dried blood, or we rehydrate it, we dilute all of those, and then if it has a lot of antibodies, if we add virus to it, that virus will be completely neutralized. But if it does not have a lot of antibodies, namely on the site where we've diluted it too much, there will be infection, and so we can read that infection because of the tag that the virus has. Now, before I really give away my main point of this whole presentation, we first ran into a bit more of a practical problem because we don't know if dried blood is stable. Serum, we've been using that for decades and decades. We know you can just pop that in the freezer and you're good. But what about dried blood? Is that the same? Does the function alter over time? Especially because we were keeping these samples for at least 5 months. We need to know that first. So we did a bit of a stability test where we spiked samples, both serum and dried blood, with 2 concentrations, 100 micrograms and 10 micrograms, and then we used serum as our control, which we put in the freezer, and we used our dried blood at room temperature, which is approximately 20 degrees Celsius, or in a freezer of minus 20 degrees Celsius. And then simply every month or so, we would measure it and see, does anything happen to the neutralizing capacity? Well, you already see it in the graph. On the left, you see the raw data, where we have expressed the neutralizing capacity in ID50 on the y-axis. This was in international units, and the storage time on the x-axis. And you see that there's a little bit of an increase in two of the sample types and a decrease in the other one. If you look at panel B on the right, there you can see it expressed as the baseline. And indeed, some samples seem to cross the upper limit of acceptance, while the other crosses the lower limit of acceptance. But that was only after 6 or 9 months. So for 6 to 9 months, these samples are completely stable at room temperature or at -20, which gives a lot of logistical freedom when we're talking about trials, which is it's already really hard. Another thing we try to do is a bit of a pilot when it comes to temperature stress. So if we were to put these samples in -20, but you have to ship them, then you still have some issues there. How are you going to get them from one country to another? Could we add an extra freeze-thaw cycle, or could we maybe ship them just, unfrozen? So we did that as well. We had two control situations, either no storage at all. We had a control where we had no stress, so these were just kept in -20. And then we had 5 conditions where we thawed them an extra time. We kept them at a certain temperature, as you can see in the x-axis, for 48 hours, and then we froze them again before the analysis. And here as well, as you can see, none of the limits of acceptance were crossed, so they were all still stable after these short periods of temperature stress. Now, really the moment suprême, the correlation between the two sample types on the left. So you see the serum on the y-axis and the dried blood on the x-axis. And honestly, I've been questioned, my scientific integrity has been questioned twice already today, talking about this, if this is the real data. It is. I really vow by that. It's a beautiful correlation, and so it means that you can use dried blood interchangeably for serum. We again checked if over time we validated our own stability test on the right side of this slide. You see that the storage time did not alter the ratio between your dried blood and your serum, so we are convinced that even after almost 300 days, you can still use your dried blood when it is in a frozen condition. So that brings me to my conclusions that neutralizing antibodies against RSV in dry blood are highly correlated with serum. They're stable for at least 6 months and possibly even 9 months if they are in a frozen condition, and they can also withstand temperature stress. That does come with a few limitations, so our stability trials were only just a one pilot study. We had one measurement per time point. If you really want to be more certain about it, then we would have to redo this. And also these measurements were mostly for an IgG immunoglobulin. If we want to know more about IgA or IgM, we would have to repeat a study because we don't know if they behave differently in the dried blood. But as Natalie was mentioning before, we should look more at impact. So what is the impact of this study? We think it's a patient-centric solution, but also a logistical solution for vaccine or immunization trials, especially in remote areas in LMIC. However, currently we are still using transportation, and you do the analysis in highly developed countries, even though I'm promoting that trials should be in the place of the highest burden. So we looked into that as well, and I'm happy to say that we have a really nice collaboration with the Noguchi Institute in the University of Ghana, and they're present here today. We have transferred the technology to the Institute really successfully, and the preliminary results you can see here on the right. There's already a nice correlation showing, and if you want to know more, I'm sure that Jude will answer all your questions at his poster or in the abstract. So that leads me to the end of my discussion. I said I would come back to how it went to take that high road, the cliff road. That was very scary. I might have been called some names. I did fall once, but I ended up being quite all right. And as you can see on the picture, we're all smiling. So it's really worth it to take that daring road. And so I am appealing to all of you who are involved in vaccine or immunization trials, whether they're ongoing or you're about to start them. Please think about using dried blood and use this as an easy solution. Save yourself some resources, but also make your patients a little bit happier. And with that, I'd like to thank the entire team from my own institute, but also the Gates Foundation and the NIH. Thank you.
Thank you, John. Are there any questions? Yes, please.
Well, thank you for a nice talk. We have done similar things for measles also, for actually exporting serum samples or blood samples actually from Africa to Holland. And the interesting thing for measles was you could also do sequencing, direct sequencing on the samples. So I could imagine that the combination there, obviously for RSV we have no proper viremia, but I still think that for other RNAs that you want to detect quantitatively, it could be done as well. And you could just put them on a filter paper and send them over. So that's an extra possibility there.
Thank you for that addition. Thanks.
Any more questions? Okay.
That was a great talk. So just to be on the funny side here, with the cliff going down, I can see what the danger associated with it. So this is great. So is there something that we should be concerned about? Like, what is the downside for this dried blood? So.
Yeah, thanks. Excellent question. So I think the major downside of it is more practically. So a lot of the times in vaccine trials, we're not only interested in the neutralizing antibodies, but you might be interested in cells, in immune cells. How does that work? And so it will not be possible to do that with your dried blood. So you are limited, but even if you could have certain time points and only use the dried blood for those time points where you only would normally take serum, then I think that would already be a major win.
Thank you so much.
Thank you.
We are perfectly in time, and so my pleasure now to invite Mary McCabe here. Mary comes from the Belfast Queen's University in Northern Ireland. She's holding a Bachelor of Science in Biomedical Science, and in the last years she's been working within the virology group in the Experimental Medicine of Belfast Centre, led by Professor Power, focusing mainly on interferon, and our research today is exactly about interferon. Please.
Okay, so good afternoon or good evening. Today I'm just going to tell you a little bit about part of my PhD work, which looks at a relatively unknown type 1 interferon in the context of respiratory viral infection. So just to start off, I have no conflicts of interest to declare. We know that RSV disease burden is highest in infants between 6 weeks and 6 months of life. We also know that the majority of kids who are hospitalized as a result of severe RSV infection do not possess any predisposing risk factors, and therefore don't receive this beneficial prophylactic treatment. We also know that RSV primarily infects and replicates in the respiratory epithelium, so it's likely that interactions between virus and epithelium play a role in its pathogenesis. And we also understand that there's maturation of the adaptive immune response with chronological age. But what we don't quite understand just yet is there maturation of the innate immune response with chronological age, especially within the context of respiratory tissue. So it's my hypothesis that a relatively immature innate immune system early in life increases your susceptibility to severe RSV viral disease. And I hypothesize that innate immune responses to infection develop with chronological age. And so to answer this hypothesis, we generated well-differentiated primary nasal epithelial cell cultures from infants at birth and those same infants at 1 year. And these cultures provided us with a window to assess the development of innate immune responses to RSV infection over this first year of life. So we obtained nasal brush samples from healthy infants at birth, and then again those same infants at 1 year. We grew these samples into our well-differentiated primary nasal epithelial cell cultures, or air liquid interface cultures, and then mock-infected or infected with a clinical isolate of RSV. We then extracted RNA and then conducted total RNA sequencing. Because of our unique cohort, we were able to look at responses in 3 categories: responses with infection, responses with gestational age, and responses with chronological age. Today I'm just going to tell you part of the story, and part of that story, regarding responses with chronological age. Differential gene expression with infection between newborn and 1-year cultures revealed a higher number of differentially expressed genes at 1 year. Now, this supported our hypothesis that we see a broader and more robust innate antiviral response to infection at 1 year. However, if we look at those genes which are solely differentially expressed with chronological age, we saw 156 differentially expressed. Now, many of these genes didn't have any previous known antiviral roles. However, some did possess innate antiviral functions, and therefore, if they have reduced expression early in life, could this increase your susceptibility to severe RSV-related disease? And in particular, we saw the increased expression of a kind of novel, not very well-known type 1 interferon, interferon epsilon, at 1 year. So we chose interferon epsilon not only because it's an interferon, so of course it's relevant in the terms of viral infection, but also because it was differentially expressed with infection and differentially expressed with chronological age within our data set. So interferon epsilon is a type 1 interferon, so it's similar to alpha and beta, and it's mostly recognized for its protective antimicrobial functions in the female reproductive tract. However, it is also present on other mucosal surfaces, including the lungs. However, unlike other classical type 1 interferons, interferon epsilon is kind of unique because its expression seems to be independent of infection or pathogen recognition receptor stimulation. And therefore, because of its overlapping yet distinctive properties, we know actually very little about interferon epsilon, especially in the context of respiratory tissue. Therefore, I set out to characterize interferon epsilon function and expression within airway epithelial cells. So firstly, using RT-qPCR, looking for the basal expression of interferon epsilon in uninfected, immortalized respiratory airway cell lines, you could see that interferon epsilon was present in all cell lines tested, with this unique type 1 interferon expression pattern further emphasized when we look at other mucosal interferons, such as a type 3 interferon, interferon lambda 1, or another type 1 interferon, interferon beta 1, which are not constitutively expressed. If we look at responses to infection, so responses to infection within our well-differentiated primary nasal epithelial cell cultures, you can see that there's a trend for increased interferon epsilon expression with infection and with chronological age, but it didn't reach statistical significance in this case. However, when we look at responses to infection with immortalized respiratory cell lines, you can see that interferon epsilon is present high both in the uninfected and in the infected cells. Again, with this unique expression pattern being emphasized when we look at other mucosal interferons which are induced upon infection. So if interferon epsilon is always there, it's constitutively expressed, does it act like a type 1 interferon in the airways? Does it induce antiviral responses? Is it protective against infection? So to go some way towards answering this question, I received human recombinant interferons and pre-treated immortalized airway epithelial cell lines with these interferons and then infected with a fluorescence reporter virus. Using fluorescence as a surrogate marker for replication, I was then able to assess differences in viral replication as a ratio of uninfected to infected cells. So using a RSV, which expresses a far red fluorescent protein, you can see the interferon epsilon in the purple. Once you pre-treat with interferon epsilon, you get around a 2-fold reduction in viral replication. And what was interesting is that this response is very different to the other type 1 interferon pre-treatment, interferon beta 1. And this response was highly dose dependent. Similar responses were seen with other viruses tested. For example, EMCV, Sendai virus expressing EGFP, and a clinical isolate or SARS-CoV-2, in this case, Delta. So these results not only emphasize the importance of interferons providing protection against infection in airway epithelial cells, but also emphasize, despite interferon epsilon's unique expression pattern, it is in fact antiviral against respiratory viruses. And finally, to determine interferon epsilon downstream induction of antiviral restriction factors, again I used human recombinant protein and pre-treated immortalized airway epithelial cell lines to assess the magnitude, but also the timing of the induction of these downstream drivers of protection against infection. And using the classical antiviral restriction factor MX1 as an example, you can see that interferon epsilon in the green possesses this unique expression pattern. Induction of MX1 with interferon epsilon occurs very, very rapidly and peaks at around 4 hours post-treatment, but this response quickly degrades. And this was similar for all other antiviral restriction factors that I tested, including IFIT1, ISG15, and Viperin. And finally, because type 1 interferons also possess a pro-inflammatory aspect, I conducted our TQ-PCR for the induction of pro-inflammatory genes IRF1 and CXCL10 to assess if, like interferon beta with type 1 interferon, is interferon epsilon capable of being pro-inflammatory in the airway epithelial cells. And again, as you can see, interferon epsilon in the green, similar to the induction pattern seen with the antiviral restriction factors, induction of pro-inflammatory genes does occur similar to interferon beta, but this response is very rapid and degrades very quickly. And so, to summarize what I've told you today, interferon epsilon expression is age-related with a higher expression of interferon epsilon in 1-year-old children. Interferon epsilon expression is endogenous, and it is capable of being antiviral against respiratory viruses in airway epithelial cells. But the induction of downstream ISGs is unique when compared to other interferons. And so to bring this back to my fundamental hypothesis, is there development of innate immune responses with chronological age? Well, I think interferon epsilon is part of the story and kind of fits into this quite nicely. If it's endogenously expressed and is capable of acting like a type 1 interferon and being antiviral against infection of respiratory viruses, I believe that if you have decreased expression of interferon epsilon at birth, this may increase your susceptibility not only to RSV, but also to other viral infections. Elucidation of age-related responses to RSV infection is critical to make sure to identify biomarkers and also for therapeutic targets. I believe interferon epsilon, because of its unique expression, its antiviral nature, but its less potent pro-inflammatory phenotype, could now be considered in those efforts. Finally, that just brings me to my last slide, to thank all the contributors who contributed to this work, from the Wellcome Wolfson Institute of Experimental Medicine in Belfast. I would like to thank my funders, and of course I'd love to thank RSVNet, who provided me with a travel grant to be here today at this wonderful conference and in this wonderful city. And of course I'd like to thank you all for listening. Thank you.
Thank you very much. Excellent presentation, excellent work. Any questions from the audience or from the former winner?
Yes, I did have a question. So I 100% agree with the idea of the innate immune system maturing over time. Because I don't know anything about interferon epsilon, but do you know what the drivers are that lead over that year of growth that increase the interferon epsilon being present? Is it due to the child encountering infections that the innate immune system learns it over time? Could you speak to that if you know?
That is a great question, which I don't know the answer to, but most likely, yes, it will be exposures to bacteria over the, microbes over the entire year of life, which I think would enhance expression of interferon epsilon. Interferon epsilon has been shown to be controlled by hormones in the female reproductive tract, but of course that's not really relevant for young infants. So it's unknown at the moment what is driving the increase in interferon epsilon, although it has been shown to be increased in children compared to adults in another study for SARS-CoV-2. So it's definitely something which is relevant for pediatric infections.
Only a comment. As a neonatologist, I see room for understanding why neonates and young infants can be susceptible not only to RSV but to several other viral respiratory infections. Obviously, I dare to say this is not the only lacking agent in the complex context of the mucosal immunity in neonates. It would be very interesting to see in vivo, not only in cell lines, whether this is an applicable issue. Do you have any experience about that, or do you plan to have any experience about that?
So I plan to continue this work within the air-liquid interface cultures, but I would love to get my hands on some interferon epsilon knockout mice to do some experiments. I haven't got them yet, but that is the plan, hopefully.
Great. Okay, if there are no more questions, we can applaud once more Mary. Thank you. And I leave the microphone and the next introduction to my fellow colleague.
So we have one last speaker for the competition today. So I get to introduce Tangen Yannick from the University of Buea in Cameroon. So please make your way to stage. I'll introduce you a little bit. Tangen is a biochemistry student pursuing a master's degree at the University of Buea. There he has focused on doing an in silico vaccine design against Pseudomonas aeruginosus and working with their advisor there on multiple projects. So welcome, and the stage is yours.
Thank you very much. Thank you very much. Okay, I'll be presenting on in silico vaccine design against multi-epitope, that is against RSV. Actually, I'm working with Dr. Shea Robert, who is also in Germany for a research work. So in silico vaccine design is a method that we use computational tools to actually screen data sets to search for potential antigens so as to design vaccines with high antigenic efficacy. RSV is a linear-stranded, single-stranded RNA virus that encodes 11 proteins. That is, it has 2 transmembrane proteins, the G glycoproteins, which is involved in binding to the cell surface, the F glycoproteins, which facilitate fusion to the cell membranes. And as you can see, that's the structure of the virus. Now, RSV causes respiratory tract infections to young children, old adults, and also immunocompromised individuals. It's a leading cause of bronchiolitis and pneumonia in infants and young children. It was estimated in 2015 about 1.5 million cases of RSV related to adults aged 65 and more in industrialized nations. Mortality in developed countries is not common, as occurring in less than 10 individuals per 1 million live births. It's a leading cause of death globally. About 1 on 50 deaths in children aged 0 and 5 years, and 1 on 28 deaths in children aged 1 and 6 months. And it is quite, I don't know how to say it. For Cameroon, we don't have any epidemiological data to assess the prevalence of RSV in Cameroon, and it's a call for concern. RSV is spread through direct contact like handshake and through respiratory droplets, runny nose, and coughing. So those who are infected are mostly children, that's young infants and young children, and older adults, individuals with weakened immune systems. Now, the control strategies are mostly supportive treatments, RSV targeted therapy, and vaccines like RSV and Abrexvo, approved by FDA, which is limited because it's costly, and provide immunity only to older adults. And we said that a novel, robust vaccines could reduce the global disease burden. The vaccine strategies for RSV include the recombinant vector-based vaccines, nucleic acid-based vaccines, protein-based vaccines, and live attenuated vaccines. Now, our strategy, what we did, multi-epitope vaccine against RSV would be an invaluable addition to current control efforts. B cells and T cell epitopes from virus proteins plus a built-in adjuvant to design multi-epitope vaccine with high antigenicity and immunogenicity. Conservation analysis to assess cross protection across different virus strains. We also did immune simulation analysis to predict elicited immune response, and 3D structure prediction and refinement to investigate interaction with toll-like receptor 4. Our objective was to computationally design a robust epitope-based chimeric vaccine against RSV. And this is our method. What we did, we obtained proteins from the Unipod database, and we did some preliminary characterization. We also did epitope prediction and chimeric construction. And we also did antigenicity and allergenicity prediction. We also predict secondary structure and physicochemical characterization. The 3D structure of the constructed chimera was also predicted and also refined. We did molecular docking and immune simulation. Now, this is the data that we got from when we did analysis on BLAST, and it showed that the vaccine, the protein candidate for the vaccine that we choose based on the antigenic property is conserved. So this is the data. We said RSV multi-epitope vaccine is antigenic and non-allergenic. And the tools that we used to analyze was antigen Pro and Vaccinen 2.0. And all of the vaccine, the proteins proved to be antigenic, and none were allergenic. We also predicted the instability of the proteins, as you can see from the slide. Now, what we did was we combined B epitopes and T epitopes in our vaccine candidate. So for the linear B epitopes, we use A, B, C pred and BP pred in order to predict the epitopes. And for the epitopes, even the epitopes that were predicted, we selected those epitopes with high antigenicity. And for the linear B epitope, we collected 8 linear B epitopes. We also did CD4 epitope prediction. The server that we used was NetMHC 2.0, and the basis it's based on affinity. We collected 7 CD4 epitopes, and cytotoxic T cells, that CD8 was then predicted using the NetCTL 1.2 server. And these epitopes that we collected, we then used to construct our chimera. So this is the design of the chimera, how it looks like. So we have the adjuvant at the N-terminal, and we have flexible linkers. This adjuvant helps to increase even though the chimera itself would induce an immune response, but this adjuvant helps to increase the immune response. So we also use flexible linkers for linear B epitope, we use KK linkers. For CD8, we use AAY linkers. For CD4, we use GPG linkers. And at the end, the C terminal of our protein, we added 6-time histidine tag. Now, this data was collected using a CSIM server, and the immune simulation showed correlates of protection. As we can see, there was antibody production and also interferon, interleukin, interferon gamma, and interleukins 2, as you can see from the graph. Now, after that, we did a 3D structure prediction using the AlphaFold server. And this, as you can see, that's the structure of our vaccine candidate. And we did validation using the Galaxy Web server. And then the protein was, the 3D structure was further validated using the POSA web. We also did molecular docking using the CLOS-Pro server. And from the data that we got from the molecular docking, it showed that our RSV multi-epitope vaccine candidate interacts with toll-like receptor 4, which indicates that it could be a potential vaccine candidate. We finally conclude by saying that the designed chimeric vaccine candidate is immunogenic, can be used as a complementary tool to achieve RSV elimination. The designed vaccine candidate showed a high affinity with toll-like receptor 4, that is, can induce an immune response. Finally, RSV multi-epitope vaccine 1 displays potentials that warrants further investigation as a vaccine candidate. So what we need to do now is left for us to do further investigation in vivo and in vitro to then investigate the efficacy of the vaccine that we just designed. Thank you.
So we do have time for questions if those in the audience have questions they'd like to ask. I can kick off one while people decide. So this type of in silico approach that you're using, is it something that you think is easily applicable in different hands and different labs all over the world to identify these epitopes to use for vaccine design?
Yeah, what I think is this method is very, I can say is preferred because you can screen large data set in a short period of time, and it is less costly. Yeah, that's what I can say. And what you do is, I think the antigen that is predicted, these tools, these online servers, these tools that they have been programmed in such a way that they have been programmed to work as though it's a human system. So most of the times, these analysis we work out based on what we reviewed before we embark on this work.
I think we have a few up here.
Hi, Keena Swanson from Pfizer. It was a very nice presentation. The approach that you use to select the particular regions of the antigens in the vaccine candidate, I think makes sense to me. Where I'm asking is from the perspective, when you are using a tandem polypeptide approach, generally you'll be evoking more, potentially more of a T cell response compared to a neutralizing antibody response. You may get both, but I think there might be a different ratio compared to different vaccine platforms. So I was just curious in your immune modeling, first of all, what were the data sets that were used as inputs into the model? And how did you use the design of the vaccine in order to make the predictions for correlates?
Now, can you, just the part of the question where you started talking about, where you started asking?
So the basic question is, with the tandem polypeptide, you may get more of a T cell response compared to a neutralizing antibody response. And we know with a lot of the data that's out there from natural history studies and other vaccine studies that it's clear neutralizing antibodies are important for protection, but they're likely not the only thing important for protection. So how did you incorporate that into your modeling relative to the design of the vaccine itself?
Okay, we did, I said we also combined not only the T cells, we also did linear B epitope prediction. So we did linear B epitope prediction and T cell epitope prediction. So combining the two, I think will create a more robust immune response. I don't know if...
Okay, I can follow up with you after.
Okay.
That's fine.
Thank you.
Maybe we can take that last one if it's quick.
Yes, I just wasn't clear how many antigens you were proposing to include in the vaccine. It seemed like quite a few, if I was reading that diagram correctly. How many antigens were going to be included overall?
Now, we use 8 linear B epitopes, 7 CD4 epitopes, and 7 CD8 epitopes.
Okay, so quite a few.
Is it clear?
Yes, thanks.
Thank you very much.
Thank you so much. That is our last presentation for the competition.
Thank you very much.
I'll turn it back over to Paolo to wrap up.
Well, thank you to all 4 presenters. It's been a great session, but most of the amusement has been reviewing the abstracts, learning very much from them. And it's regretful that the additional 47 couldn't be displayed, but you can see everything around here in the booklet and in the poster exhibition. Now, I ask the organization to display the QR code because all of you are strongly invited to vote for the best presentation and the best science among these 4. You have time to vote until 11 a.m. on Friday. You can vote through this QR code. Please do it. We'll be announcing, Ashley and I, the winner in the final session on Friday afternoon. And so Ashley will know who will be the one replacing her next time. So with this in mind, I leave the microphone once more to Ashley. It's been a pleasure to chair this session together with her, and I wish you a fruitful and very interesting follow-up of the meeting.
And I'll just say thank you for all attending, and to reiterate to stick around to the end of the conference where you will see the winner be announced. And those of you that have trainees and students, encourage them to apply and to compete in the event for the following years to come. And thank you to the speakers that, got up on the stage to talk about their research today. So thank you.